Dear participant, we are with Dr. Celia Chalam, madam, and as madam has taken lecture on national quarantine system, plant quarantine system. I think I should not take much time to again introduce because although madam has been introduced by earlier. So, and madam has a vast experience in virology. So, I request ma'am to share your slide and please give your word of experience and share your experience with our participants. Please, ma'am. To you, ma'am.
Thank you, Dr. Subhash. As earlier, I have already taken the practicals. What I will do is, I'll just give a glimpse of to make the story, how it is done in virology. Then the respective practicals will be taken by Dr. Pooja Kumari, scientist, and Dr. Kalai Ponmani, he is a project scientist 3, and then Ms. Priya Yadav. So in quarantine, what we do is, as I have explained earlier, we just give me a second.
Presentation mode, ma'am.
Yeah, I'm carrying. Is it done?
Note, ma'am.
None. Okay. Yeah, the quarantine, I'll let it open. I'll continue instead of wasting time. We have a station at headquarters, Delhi, and also in Hyderabad. And the transgenic germplasm quarantine is done only at Delhi. And this is how the processing starts. We look for the documents, whether import permit is there or not, and phytosanitary certificate given by the exporting country. And then we maintain the register. And this is how the samples are recorded. You can see here IQ number, it has come from Taiwan, tomato, and 5 samples. And 5 samples were released after giving prophylactic treatment with trisodium orthophosphate. This is for tobamoviruses. And then the release date. And this is the joint inspection by pathologist, entomologist, virologist, nematologist, weed scientist. And then all the symptomatic seeds are removed. And this is the avocado imported from Australia. And I just take you through the virology and all the symptomatic seeds like this. These seeds are soybean infected by soybean mosaic virus or bean pod mottle virus, not reported from India. And broad bean seeds infected by broad bean stain virus. And then the pea seed borne mosaic virus, pathotype 4 is not there. So all the healthy looking seeds goes for post-entry quarantine growing. And all the legumes, whether you are importing peas, soya bean, faba bean, black gram, green gram, all of them goes to post-entry quarantine growing. And this is the post-entry quarantine facility what we have in Delhi. And you can use both the soil-based greenhouse and the benchtop greenhouse. And this is, and also the samples is released and undertaking to grow them at the indented site. And our scientists go and then collect the samples. You can see here the tomato infected by pepinomosaic virus. And this is maize. And all the symptomatic plants, we do the electron microscopy. Dr. Pooja will explain you. And we do the ELISA. We have 50 to 70 exotic viruses antisera. And Ms. Priya Yadav will explain you. And this is the flow of practicals we will be showing. And then Dr. Kalai Ponmani will show you how the reverse transcription PCR is done, because all the seed transmitted viruses are so far RNA viruses, and recently some DNA viruses also found to be seed transmitted. And Dr. Pooja will explain you about the DNA viruses PCR. And we have intercepted 45 viruses, including 19 exotic viruses. This I have shown you during the theory class. And during post-entry quarantine inspections, wherever the symptomatic plants are there, we uproot. And if the state government permits incineration, we incinerate them. Or if they don't have the provision, no provision for incineration because of pollution issues, then we bury them in the pit and destroy them. And the transgenic germplasm, the procedure is same, except that we do the entire thing in containment facility with high efficiency particulate filters and with the RCGM clearance. These were explained to you. And transgenics also, we have intercepted many viruses. And whatever that is grown either in the containment facility or in the post-entry quarantine greenhouses, non-transgenics, everything is destroyed following all the standard operating procedures. And you can see here the incinerator, what we have in NBPG. And this is what is done at the indenter site. And at the end, once all the tests are done by entomologists, nematologists, pathologists, and weed scientists, then the material is released to the indenters in a pest-free state. In our case, it is a virus-free. So some of you have not attended the theory class. That day also only 40 participants were there on Saturday. That's why I took you through the whole story of how the quarantine processing is done. And I'll start with the post-entry quarantine, then Dr. Pooja will Dr. Kalai, you are sharing the screen?
She's asking. That also. Okay. Showing man. You can ask this one. Just about click on it. This part.
Have the video also post-entry quarantine embedded in this?
No, ma'am.
Then close this. I will just explain the post-entry quarantine and then Dr. Pooja can take over.
Shall I stop share then?
You stop sharing.
Shall I share that video, ma'am? Or you can?
Let me try.
Fine.
Yeah, easy for me to explain. Could you see that video?
Yes.
Yes, ma'am, visible.
They have not inserted this.
This is if you are doing the in-person training, you would have come here. This is NBPGR. the post-entry quarantine facility what we have, and this is the one, the containment facility. Yeah, this is the post-entry quarantine facility what we have. We have Dr. Pooja Kumari here and Dr. Bharat Gawade, nematologist, and Mr. Ashok Moria, technical officer. And we have Mr. Satyanarayana, who is the one actually takes care of the all the facility. He is a very good support for us. And the post-entry quarantine facility should have the double door. This is the first one. Yeah, then the second one, and we have all the controls for temperature, humidity. Yeah, this is the all the vents should be closed with the 40 to 60 mesh net. You can see you have the fan outside, but this is covered with the 40 mesh net. And here we have grown soybean. And this side we have coffee. Yeah, each accession is grown in 1 or 2 rows. You can see the accession numbers here. And then as and when they grow, they show the symptoms. So we collect the symptomatic leaves. I'll just fast forward the video because the time is less. Yeah, these are some of the symptoms like mosaic. So these samples are collected. You can see the puckering symptoms here. These are the virus symptoms. So these samples have come from USA and they are showing the symptoms of mosaic. You can see the very good puckering here. You can see. This is the yellow mosaic symptoms. And the Dr. Pooja is collecting the samples. And also we collect the representative healthy samples because viruses are known to have the latent symptoms. So we test both the symptomatic and also representative healthy looking samples. Why I am saying healthy looking? These are mosaic symptoms, yellow mosaic symptoms, and the healthy looking, why I am saying is they look absolutely healthy, but when we test them, we find the viruses. So this is the benchtop greenhouse. So you can grow the samples either in the soil-based greenhouse or benchtop greenhouse. I'm just fast forwarding it because we have to show you some more videos of electron microscopy. And also the post-entry quarantine greenhouse has head house to prepare the soil. And we will take you to this is the incinerator what we have. And then all the material which is grown inside the greenhouse is incinerated here. Dr. Kalai?
Yes, ma'am. I am sharing.
Yeah. This is how the post-entry quarantine growing is done. And then Dr. Pooja will take you through the electron microscopy infectivity test, and Priya will take you through ELISA, and Dr. Kalai Mani will take you through the PCR.
Once you sit here, you start video and then you can ask.
Okay, whether presentation is visible on your screen?
This is Pooja.
Okay, okay, fine. Next. Responding.
Most of this down arrow.
Okay. There is some problem again. It's not responding. It is blinking also. Blinking. This cursor. Just please wait 1 minute.
Can you the scroller
Stroller.
start video and then just scroll.
So basic introduction part, everything covered by madam. And this is, I will just show you the idea like how we are conducting this biological method for virus detection. So based on symptoms, actually infectivity test is performed. Once again. So as regarding this infectivity test is considered, so we collect the symptomatic virus symptomatic or infected plant sample, then we crush it with the help of piston motor and prepare the extract using the phosphate buffer. Then the test plant we select. In the test plant we rub this infected sample. First we use slide or carborundum as an abrasive. After creating the rupture, then we rub the leaf abstract on the test plant. Then we remove the inoculum, excess inoculum with the help of this sterile distilled water. Then we observe the symptom in the test plant at 1 week or 2 weeks interval, like what type of symptom it is producing, whether it is developing a symptom of mosaic, mottling, and leaf narrowing. Okay, problem actually. Scroll and do it. It's showing problem again. Again, this scroll is not.
Dr. Khuda, is the screen okay for you?
Yes, ma'am.
Full screen?
Yes, ma'am.
Yes, ma'am.
Screen is
Okay,
all right.
then. Dr. Pooja, carry on. Maybe some issue
But
with
his
my...
language, language is hazy. It's
Ma'am,
not readable.
actually, this here our presentation is not moving. Okay, now. So because of this technical problem, no,
Sorry.
no issues. Okay. So this is the pictorial view, like as I mentioned verbally earlier, like this is the symptomatic plant. We prepare the sample extract using the phosphate buffer. Then through the carborundum or slide as abrasive, we rub it. Then we we just spread the sample inoculum, then washing with the water. Then we keep this test plant. It's moving. No? Then we keep this test plant for symptom observation at weekly interval. Again, problem it is showing. Not moving.
Can you click this slide? Click this slide and then forward.
Actually, sir, I'm trying, but maybe due to network issue, it is not moving in my screen actually.
Unshare and share.
Can you unshare and then again?
Unshare and share.
Yeah.
Yeah, now it is moving. Go ahead.
I think now I think more clear than earlier. Yes, I think it's moving now.
Dr. Pooja, it is moving. Carry on.
The slide is moving, but the fonts are hazy.
Fonts is hazy. It's not readable. But I think you continue.
We finish it, no? Video and everything. This is physical matter.
This is
Now it's
fine.
okay.
Carry on, please.
Yes, ma'am. The physical method, earlier I covered this biological method of examination using infectivity test. Now physical method for virus visualization using electron microscopy. So electron microscopy, we know the basic fundamentals. So just I will show you the methodology, how we are conducting. Again, it is creating problem. It's not moving. It's not moving. So since this slide is not moving, just verbally I will explain you methodology, how we are doing it. Actually, we collect the symptomatic virus symptomatic leaf sample, and we just crush this. We take the two very small size of the infected leaf, and we crush using this 0.07 molar phosphate buffer in the glass slide using the glass rod. So using phosphate buffer. So once the sample is finely crushed, then we use here the copper grids. So copper grids are used for, then we just with the help of this fine tweezer, we just take it out the copper grid having the 400 mesh size, and we place the extract, the inoculum what we have prepared on the grid. So we keep it for drying for 2-3 minutes. Then afterwards, to remove the excess inoculum, so what we do, we use the sterile distilled water to remove the excess inoculum from the grid. So afterwards, we use the filter paper to just wipe it out wherever just droplets are there around the grid. Then with the help of this tweezer, we do here negative staining using this 2% uranyl acetate as a staining agent. So we drop by drop, we place this staining agent on the grid. Then we keep it for drying. So here this staining is called the negative staining. Why we call it? Because the background take the stain here. Therefore, it is called negative staining. Then this after once this grid is dried and treated with this uranyl acetate stain, then we hold this grid using the tweezer and we place it in the EM. One holder is there in the EM equipment. So in the holder we place it, and then we insert this holder inside the EM, and accordingly we visualize at different magnification, like generally we follow 80,000 or 1 lakh magnification. We visualize in the fluorescent screen the presence of virus, and here EM give the idea like which shape of virus is our test plant or infected plant having, what type of virus infection, whether it is of rod shape or spherical. So this size and shape idea we will get it through the EM. Actually I am waiting, but still this slide is not moving.
Dr. Pooja, unshare the slides.
Okay, ma'am, just I will unshare. Stop share.
You can directly start showing the
And then show only electron microscope video separately. Don't,
There is a problem in the system. So video is visible?
yeah, go
Madam.
ahead.
Yes. So as verbally I explained, the same thing I will repeat here. You just have a look like the symptomatic leaves are collected, and he is the chief technical officer. He is regularly do the EM part here in our laboratory. So this is the symptomatic leaf. Small portion he will cut it. Then this we have placed it in this glass slides. Then this is the phosphate buffer, 0.07 molar. Strength phosphate buffer is added with the help of this glass rod. This fine crushing of the leaf sample. Then this is the holder having this copper grids, which is carbon coated. Then this is the fine tweezer. With its help, he will just pick the copper grid. Where sample will be placed. Then just is loading the sample with the help of pipettes, then placing on the carbon coated copper grid. Then allowing it to dry for 4 minutes.
You can start presenting video.
Then washing with the sterile distilled water to remove the excess inoculum and plant extract, plant debris, everything. Just washing with this water. Then this is the uranyl acetate for staining it used. So drop by drop it is placed on the grid. Then just drying with this blotter or filter paper.
Dr. Pooja, can you please fast forward the video?
Okay, ma'am. Then
Because
now
we
this
have-
grid is ready. This is having the test plant sample, so it is loaded in the holder. Then the just holder is inserted in this EM machine. Then it's visualization in the fluorescent screen. The magnification is setting at different magnification, doing proper adjustment. Then visualization of this grid. This is the adjustment. So in that screen, virus shape and size, everything will be recorded in the computer screen. So this is all about this EM.
From there, you can just ask.
Okay, now Priya will explain the protein-based virus detection, that is ELISA, using ELISA method.
Yes, sir. Explain.
Priya, you are muted.
Priya, you are muted, I think. Yeah.
Your madam, your screen is not showing anything.
I'm sorry, it was muted.
हाँ जी, हाँ जी, बताइए.
Shall I speak again?
अच्छा, मैं फोन करके बात करता हूँ.
Yes, yes. Priya,
Screen
you
is
are
ठीक है, ठीक.
already in
बोलो,
sharing
बोलो.
mode. It is not coming, ma'am.
Yes, screen shared is not showing anything. Unshare your screen first and then again you share, madam.
Oh God.
तो कितना देर लगेगा पहुंचने में? पहुंचे नहीं आप?
Sorry for the network glitch. Actually, due to network issue, we are unable to go through the PPT. So I'm directly presenting the video. So as I was telling, ELISA is the enzyme-linked immunosorbent assay, which is a serological technique used for the detection of an antigen or antibody. Here in our lab, what we do, DAC and DAS ELISA, and I'm showing you the video of DAS ELISA. DAS ELISA is the DAS ELISA is the type of direct ELISA. Uh, Here we can see the first step is antibody coating and washing. What we do here, we take anti-sera like and capture antibody and mix with coating buffer uh, in the ratio of 1:200, and then we mix it properly. And after mixing, we use 100 microliter of the uh, capture antibody and load it into the ELISA plate, which is polystyrene plate and has inherent capacity to bind the protein. After loading antibody, what we do keep our ELISA plate in a plastic bag, and later on we incubate at the temperature what temperature is required, as different company different companies having different protocols. So accordingly we follow the protocol and keep for incubation. Here we are keeping, after keeping our capture antibody along with the ELISA plate at 4 degrees Celsius. After incubation is completed, we take that box out of the fridge and then we start washing. We use PBST buffer for washing. Here we see the leaf sample, to which first of all we weigh and then we grind it by using mortar and pestle along with sample preparation buffer. After sample is crushed in a sample buffer, we centrifuge that sample at some 4,000 RPM for 5 minutes or 3,000 RPM for 5 minutes. And then we take out the tubes and transfer the supernatant into new tubes. Then we load the samples and along with the positive control, buffer control, and negative control. And then again, we keep ELISA plates in a moist box uh, at 4 degrees Celsius for overnight. Once incubation is completed, uh, we take out our moist box and ELISA plates, and we start washing. After washing is done, the third step is the conjugate preparation, loading and washing again. Here we can see the conjugate enzyme linked alkaline phosphatase conjugated antibody, and we dilute it in the same way as we did in case of capture antibody, and load it into the wells. And here we use conjugate buffer, as there are different buffers for each step. As we saw in case of sample preparation buffer, we use a sample extraction buffer, and in case of coating antibody buffer, we used coating buffer, and here we are using conjugate buffer.
Pooja, Priya, please fast forward and go to the fourth final step because we have only 10 minutes more.
Voice was not coming.
No, go to the fourth step, I am saying.
Don't stop. Speak. Just go with the flow. Video flow.
Okay,
Fast
okay.
forward.
After conjugation step is completed, after conjugate step, there is the last step, substrate preparation and substrate addition step. What we do here is, um, we take PNPP substrate as we used alkaline phosphatase. So for alkaline phosphatase, we use PNPP substrate, and here what happens when we add PNPP substrate in the sample, in the ELISA plate, it reacts with alkaline phosphatase enzyme, and it gets converted into para-nitrophenol, which gives yellow color, and it also indicates the positive result. These are the PNPP substrate tablets we use. What we do PNPP substrate tablet, and then we crush these tablets completely, and we need to perform this step in dark, because this is the After PNPP substrate preparation, we load in our ELISA plate, and then we cover it by foil paper. After everything is done, like all the 4 steps have been completed now, and ultimately we take plate reading after the incubation of half an hour time when we add our PNPP substrate. This is the Magellan 6 software we use of Tekken. Here we keep the plate. And here we can see these black color boxes indicates buffer control, and these green color boxes indicate negative control, and these red color boxes indicate positive control. The interpretation of results is done by visualization also, but here what we see, if the OD value is twice the mean of negative value, then the sample is considered infected. It was all about ELISA, and our apologies as here we have network issue, so we couldn't present so well. Now Pooja ma'am will explain about PCR.
Unshare. Dr. Pooja, if you don't have
It is visible. So since Priya has covered this protein-based detection of virus using ELISA, dash ELISA, and DAC ELISA method, now the another more reliable and very sensitive method is nucleic acid-based diagnosis for the virus. As we know, viruses have either RNA or DNA. For DNA viruses part I will cover, and the RNA virus part detection Dr. Kalai will cover. I am not moving.
Dr. Pooja, we have only 5 minutes left. Let Dr. Kalai show the video.
Dr. Pooja, can you hear us?
Dr. Pooja?
She is not audible. That Pooja is not audible.
So this slide is visible now?
No,
Yes.
not clear. It's not clear.
Slide is visible,
In the
but
nucleic
font is
acid-based
hazy.
But
detection for
this
the
text is not clear. No.
we are using DNA isolation. So the procedure is first the infected plant tissue 1 mg we weigh it and we crush it using liquid nitrogen. Then we add this 1 ml of lysis buffer. So once the lysis buffer is added, the suspension is entirely transferred into the Eppendorf tube. Then we place this tubes in the hot water bath at 60 degrees Celsius for 1 hour. So after the incubation, we keep it at room temperature for 5 minutes. Then we centrifuge it and discard the pellet part. Then RNase treatment is given to remove the RNA molecules present in that tube. Then we do the washing with the help of chloroform isoamyl alcohol. So based on the plant tissue, washing step varies. Sometimes it is 2 times or 3 times. So after washing, we do the chloroform treatment. Then finally, for the once this nucleic acid like DNA pellet is there in the append-off, then for its precipitation, we use the cold isopropanol to precipitate the DNA. Then to loosen this pellet in the append-off, we do the ethanol washing. Ethanol washing we do. Then followed by this finally, after ethanol washing, to remove its smell, we keep the append-off tube outside, sometime inside laminar and even in the laboratory to dry it for a while. Once it gets dried and there is no more any ethanol smell, then this pellet is dissolved either in 1xt buffer or sterile distilled water. So actually in our lab, basically for begomoviruses, we are using these sets of different generic primer, which in target some these sets of primer for DNA A molecule, some are for DNA B molecule, and the satellite, beta satellite, alpha satellites. It has the different annealing temperature and amplicon size. So a molecule OI set of primer is used. Its annealing is 55 degrees Celsius and amplicon size is 1.2 kb, and remain PCR principle is remain same for any of the pathogens or microorganism, that is denaturation at 94, then extension at 72 degrees Celsius. And this we keep this reaction in the tube for 35 cycles. Okay. Then this is for the satellite molecules. For example, here I'm showing for the beta satellite, annealing used is 59 degrees Celsius, and it gives the amplicon size of 1.2 kb. Here example I have quoted for okra initial leaf curl virus. These are the symptomatic wild okra in the field showing the yellow vein symptom. And this is the gel picture. So 1.2 kb amplification we got for these are symptomatic wild okra accession in the PCR. And these are the few publication for yellow vein. And this is for the initial leaf curl, like twisting of this branches and the small leafy growth in the lower abaxial portion of the leaf. So here this beta satellite set of primer is used for the detection, and it gave amplification of 1.3 kb for the symptomatic. And this is the publication for okra initiation. Now the DNA part, nucleic acid-based detection I have covered. So RNA, for RNA viruses, Dr. Kalai will explain you.
Subhash, this will be, we are coming to the end. Okay.
Okay.
Ma'am, from there you can unshare.
Dr. Kalai, do it fast, please.
Yes, sir. I am start sharing my video.
Okay.
Very good morning to everyone. Now I'll be showing a small video on detection of viruses, mainly RNA viruses, through RT-PCR. And here is the short video on detection of RNA viruses through RT-PCR. In our lab, we use kit, this RNA-EC Plan Mini Kit for isolation of RNA, and these are all the components of the kit: 2 buffers, 1 lysis buffer, 2 wash buffer, and butter. All these experiments will be carried out under has to be carried out under a controlled environment. We use a hood, RNA isolation hood, for isolation of RNA, and here the RNA hood is cleaned, and then sample preparation. The next one is sample preparation, where we isolate RNA, total RNA from the target sample using lysis buffer, RLT buffer. Once the sample is ground, will be transferred to a shredder column. This is the shredder column where lysis will be enhanced, and immediately after shredding, it has to be centrifuged, and the sample is now centrifuged for taking supernatant, and then the supernatant is mixed with ethanol for mixing supernatant as well as isolation of cellular component. And immediately after mixing with ethanol, the approximately 650 microliter of sample will be transferred to RNA isolation column, and then centrifuged for a brief moment to separate the supernatant. And after that, the supernatant is washed with wash buffer, 2 wash buffer, RW1 and RPE1. After the washing of sample, the filter will be transferred to a new collection tube where the RNA will be eluted with water. And immediately after elution, cDNA is synthesized. Here we use thermoscientific kit for cDNA synthesis, and this is the cDNA synthesis followed by the RT-PCR of target virus. This is the RT-PCR detection of a raspberry ring spot virus, which is a quarantine significant virus, which easily spread through vegetative planting material. Here we are showing the analysis of gel using designated software gene system, and here you can see the gel picture. And once after taking the gel picture, the gel will be analyzed for target virus and all. So this is how we will detect RNA viruses using RT-PCR. With this, our presentation is over with respect to detection of viruses, and now I request ma'am to take forward. Ma'am.
So this is how the
Others,
virus.
please mute. Others, please mute.
Dr. Kallai, please unshare.
Yes, ma'am.
Yeah, this is how we do in some of the presentations took a lot of time. We apologize for that because of the internet issues. And with this, we conclude the virology practicals. And thank you, Dr. Subhash and all others.
Thank you, madam. Dear participant, quickly we can take 1 or 2 questions. If you have any, you can ask. Anyone, please, so that we can start
Please.
another practical. Anyone? If no, then ma'am, may I propose a vote of thanks. On my behalf and on behalf of organizers, I thanks to Dr. Celia Chalam ma'am, Dr. Pooja Kumari, Dr. Kalaimani, A.K. Moria ji, and Priya Yadav for showing a practical on a quarantine processing in respect of virus detection. Madam, you all beautifully explained virus detection using PCR, different techniques like PCR, ELISA, and electron microscopy using videography. Ma'am, you all have shown post-quarantine facility of our NVPGR through videography. That is very well you've shown. And again, I thank you all for this practical, ma'am.
Thank you, Dr. Subhash.
Now, is Dr. Jamil sir, are you there? Pardeep and Jamil sir. Dr. Pardeep?
Yes, I am here. Good morning.
Okay. Welcome, sir. Welcome, Dr. Pardeep.
Thank you.
Welcome.
Thank you, sir.
Sir, we are late. Sorry for.
No issue.
No worries.
Okay. Then I should first introduce our today's speaker, and they are presenting on the plant quarantine processing in case of plant pathology. I will share the slides quickly. I formally welcome to both of you, sir, Dr. Jamil and Dr. Pradeep. I quickly introduce them. Dr. Jamil is a principal scientist, plant pathology, and they are working in division of plant quarantine in NBPGR New Delhi. And their nature of work is they have tested around 3.5 lakh imported germplasm samples, and among these samples, they have significantly interrupted of quarantine pathogens such as dipothy, longi, cola in case of sunflower and soybean from USA, and Fusarium oxysporum species, this curcumarium in melon from USA, like this, and he also reported some fungal pathogens on different crops from India also for first time, such as a perfect stage of Fusarium graminearium on rice seed, and Nemenosporium americanum in case of Agilops. And he also developed and modified several diagnostic techniques such as a detached non-wounded fruit inoculation technique in chili, and a technique of obtaining a pure culture of fungi from crop seeds, and several others. Dr. Jamil has published more than 100 research paper, and he has written book 45 book chapter, 2 book edited, and 15 technical article, and 43 popular article he has published. And there are a long list of award and recognition. Of only the few of them I am mentioning here. He has received Young Scientist Award from UP Council of Science and Technology, Government of Uttar Pradesh, and Srimati Kaveri Sharda Memorial Award by Plant Protection Association of India, Hyderabad. He is nominated as a editor and Indian phytopathology, and he is a fellow of Indian Society of Phytopathology and Indian Society of Pulses Research and Development, and the Society of Plant Protection Science. And now I introduce our another speaker, Dr. Pradeep Kumar. He's a scientist, senior scale in Division of Plant Quarantine in the NBPGR New Delhi. With the 9 years of professional research experience in molecular diagnostic of seed-borne pathogen, he has contributed through several in-house as well as externally funded projects. He has molecularly diagnostic protocol were developed for seed-borne fungal and bacterial pathogen. He has done genetic diversity analysis of seed-borne fungal pathogens using various molecular markers. And he has done plant biosecurity through quarantine processing of more than 500 lakh samples of germplasm. Seed health testing he has done in germplasm more than 1 lakh, and received received for National Gene Bank in our NBPGR. He also screened the germplasm against various pathogens. He has published more than 40 research paper in reputed journal, and 15 book chapter. He has written 15 more than 20 popular and technical article he has published. He has received a Young Scientist Award, Best Oral Presentation, and Best Poster Award in international conferences. And I welcome you all, sir. You too, sir. Now I request you to share your screen, and you do the practical session. Up to you, sir. Jamil sir and Pradeep sir.
Thank you, Dr. Subhash. Thank you very much.
See the other videos, how it appears. Huh? You keep login. Okay.
Jamil sir.
Yes, yes, I'm just sharing. Is it visible?
Yes, yes. Yes, sir.
Display mode. Good morning, friends. I'll not take much of time. I'll directly come to the detection and identification part rather than going in briefing the importance as all. Okay. So coming directly to the methods for detection and identification, especially for the fungi and bacteria. I have categorized into 3 categories like non-incubation method, then incubation and molecular, that is the advanced methods. So under non-incubation method, usually most common one is the visual examination, then the NOS soaking, then KH, washing test, embryo count, UV light. These are some of the common ones. Whereas under the incubation, the variants of the incubation methods like the blotter test, agar plate 2,4-D, seed washing test, then the growing out sibling system. And apart from these advanced methods like the molecular one, that includes the polymerase injection, real time, RT-PCR, nested, bio, and DNA chain. Okay. So these are the general features related to the different methods, how they are sensitive, how they are easy, and what are the specificity. So under non-incubation methods, usually the sensitivity as well as the specificity is low, but certainly it takes a minimum time somewhere, sometimes maybe 5 to 10 minutes. And other one is the UV light examination. Okay, then it also takes hardly 5 minutes, whereas the washing test, it takes around 30 minutes. The whole embryo count method, it takes around 24 hours. Okay, but more or less the sensitivity as well as the specificity is generally low. In some of the cases like embryo count, it is moderate, whereas in incubation method, the longer period is required, maybe from 24 hours to 3 weeks, depending on the method what we are using. Like blotter test, it takes 1 hour or 1 week. The sensitivity as well as the specificity is moderate to high. Okay. Then if I talk about the NOH soaking, it takes around 24 hours. There again, the sensitivity and the specificity is moderate to high. These are some of the advanced method apart from molecular biochemical assays like polymerase chain reaction, real time, and these all, it takes maximum 5 to 6 hours, but their specificity as well as the sensitivity very high. At the same time, they are not very simple, they are complicated and expensive too. Okay. Now, coming to our methodology, which we follow in our quarantine processing for imported germplasm. So this is the overall like joint inspection. We start with the joint inspection. There we do the visual examination, then taking the help of sometimes the magnifying glasses, depending on the crop and seed and pathogen as well. Then coming to the blotter test, then the washing test, then NOH soaking, KOH, grow out, and PQ inspection also. And ultimately, we try to detect and identify the pathogens which are associated with the imported seed material. Now, coming to the visual examination, very simple. We sometimes take the help of the magnifier or the stereo binocular. And what we observe that the infected or the infected seed, the fungal growth in the seed surface, as well as the admixture of the fungal fructifications. These are some of the typical symptoms which one can observe during the visual examination, like white streaking caused by the Fusarium verticillidis in maize, the downy mildew of soybean, the white incrustation of ooze spores that can be visualized under the compound microscopy as the golden colors, 3-layered ooze spores. The 2 important pathogens of chickpea, they are the Ascochyta rabi as well as the Botrytis cinerea, and can be very easily identified. Apart from these, some more like the bacterial infections in beans, like the halo blight as well as the bacterial wilt, you can see very well under the UV light that can be fluorescent coloration can be visualized. Whereas in case of pink discoloration of wheat, the Irvinia repnotici is the one which produces the pinkish color on the seed. Then the one very typical example of the eye-shaped spots on the wheat caused by the bipolar sorokiniana. Then the ergot, another one, the visual examination. The purple stain in very common in soybean. These are all under the visual examination. And few more like the kernel burnt, the hill burnt, then bajra smut, then the paddy burnt, then long and short smut of jowar, then the smut of jowar, then the foxtail millet smut of maize. These are all can be very easily identified during the visual examination. So I am going to just play a small video. You can have a look, please.
Plates and seeds are washed carefully for the presence of the soil particles, plant debris, insect, weed, and discolored deformed seeds.
Sir,
So for the
Video is
Not
not
visible.
showing.
sir, play again, sir. Jamil, sir,
Yes.
video is not showing, sir.
What happened, doctor?
Video
Sir.
is not.
Sir, please unshare first and then again you share and play video.
You mean video is not playing?
Visible.
No, not playing, sir. Not
Okay.
showing here.
Let me try again, and if not, I will just verbally tell. Okay. Is it okay? Is it okay, Pradeep?
No, sir.
Okay, leave it. So
Dr. Jamil, can you
Yeah.
play the video separately, not the embodiment? No, then it will work.
madam, what are you saying?
Sir,
Open
you
from-
separately play that
open
video,
directly.
sir.
Okay.
Then it-
Okay, let me check whether can I. Separately.
plates and seeds are washed carefully for the presence.
Then do I need to stop the PPT then to
Yes,
start?
sir.
Okay.
Stop it and then
Okay.
again share, sir.
Okay.
Soil particles,
Is it okay now?
insect,
Yes, okay.
and
Okay.
discolored
Yes, okay, sir.
So for the methodology plates and seeds are washed carefully for the presence of the soil particles, plant debris, insect, weed, and
Sir, voice increase कीजिए थोड़ा.
seeds.
What happened, Dr. Subhash?
Voice is very less.
So better to stop this one. Okay?
No, sir. It's okay.
Because I cannot increase the volume.
Okay.
It is the
You
maximum.
explain, sir, in video.
In video, you explain, sir.
Okay, these are in fact the symptoms. Based on these symptoms, we can easily identify the pathogens associated with the seeds. Like this is the coax smut. Okay, smut of coax. Okay,
Yes, sir.
the entire seed is converted into the spore ball. Okay, so this is the example of ergot. This is the example of kernel smut. That is the Trellissia barkiliana. Okay. So you can see over here the seed coat is split and a number of teliospores are arranged on in a line, streaking line manner on the seed surface. Now this is the purple stain of the soybean. You can see the purpling of the seed. It is due to the Cercospora kikuchi in case of soybean. Okay. Claviceps. Yeah. This is the ergot, bajra ergot. Very important. Now, yeah, this is the sunflower rust and very easily can be detected under the stereo binocular. Okay, this is the oat ergot. Okay, so, let me unshare. So. Is it visible now?
Yes, sir. Very much visible, sir.
So I'll not play the video because it is creating a problem, I think. So with this method washing test, we usually try to detect and identify the spores of the rust, especially in case of safflower and sunflower, as well as the beet rust. So usually, one more pathogen, Pernospora mansurica, that is also detected through this washing test. But usually we use in our lab the microscopy, that is easier. Okay. So here the iodospore or the teleospores can be separated out using the centrifuge. 5,000 RPM we run in a test tube having a small quantity of water with a few seeds and a pinch of sand is taken in the tube. Then for 30 seconds, we stir it on the centrifuge. We take out the supernatant. Then under the compound stereo binocular microscope, we try to identify the teleospore or the iridospore. As you can see over here, these are some of the pictures which we have captured during our examination. Like teleospores of the sunflower, it looks like English 8, okay, digit, numeral digit 8, whereas the iridospore is simply spherical, okay, single walled. Whereas in case of safflower, you can see the teleospore is something different than the teleospore of the Vaccinium helianthine. And on the seed itself directly, we can visualize under the stereo binocular microscope. Okay. Now coming to NOS soaking test, very simple. We take around 100 seeds usually because it's very easy to calculate the percent infection in the seed sample. Okay. So what we do, we take the 2 gram of NaOH in 1000 ml of water. Okay. That is how it is 0.2% NaOH solution. And the counted seeds are soaked for 24 hours. Okay, in a beaker, in a room temperature simply. Then we drain out the solution and we put the seeds on the blotter and separate them. And we count the total seeds which are converted into the jet black color. And these jet black colors are the bunted seeds of the rice. And to confirm the identity or the association of the Teresa barkleana, we tease the seeds and make temporary mounts so that we can confirm that yes, whatever is discolored, black colored it's nothing other than the Telesia barkleana. And we count those seeds and out of 100, we calculate the percent infection. This is how we do the NOH soaking for rice. And this particular pathogen is though present in India, but its distribution is very restricted. That is how we, now coming to the whole embryo count method, though it's very complicated, it's not easy. Okay, but certainly this is the one method without going for the seedling symptoms, we can detect the presence of the blue smut pathogen in the embryonic region of the wheat seed. And its specificity is comparatively better than the visual examination. And this method was developed by Hewitt as well as the Agarwal in early 1970s. So here what we do, as per the standard method, we take the 1000 seeds and in the sodium hydroxide solution 5% containing agropyran, trypan blue, 0.15 gram, and we soak for 24 hours, okay, at room temperature. Then we take out the seeds, soap seeds, then we wash under the 10 mesh sieve, then we take out the remaining into the 20 mesh sieve, then they collect the embryos, extracted embryos. Now we come to the hydration. Then during the dehydration, it takes around 5 to 10 minutes. Then we separate out those extracted embryos using the lactophenol, followed by again washing to remove the chaffy and all. Then the clearing, it takes around 2 minutes. After boiling into lactophenol, then we take out the embryos and arrange them on the measurement slides. Then if you see under the stereo binocular, all those pink color or dark color are the spotted one. These area are the invaded areas in the embryos, which can be very easily magnified, and you can see thread-like structures are invaded in the embryonic region of the seeds, that wheat seeds. This is how, and we count the number of infected seeds, the embryos, and out of the total one, this is how we calculate the percent infection. Now coming to KOH test, just to confirm whether the associated or the detected bacterium is gram negative or positive, very simple. We use the KOH solution, and we take just a pinch of bacterial fresh colony, and with the inoculation loop, we try to just make a suspension. While doing this, the cell wall is dissolved. If it is dissolved, then you can see the thread-like structure can be created when you will lift the loop. If there is no dissolution of the cell wall, it's simply a gram positive, whereas wherever it is dissolved, it's a that loop will bring a thread-like structure. Okay. Now coming to blotter test, one of the most common, and what we actually use, that is the modified ISTA method. And the observation period, that incubation period is for 7 days, and the temperature which we is maintain 22 degrees centigrade for 7 days, and the alternate cycle of light and dark for 12 hours. And after 7 days, we take out the samples, then we observe under the stereo binocular first. Then if any further assistance is required, we take the help of the compound microscopy. Even after that, we are not able to species level identify, then we go for the PCR based identification after sequencing the ITS and other regions. So already discussed. So these are some of the very important and uncommon kind of pathogen on different hosts. Like you see the Dimnosporium americanum. This particular pathogen is not reported from India earlier, and we have detected this one on the Agilopes, then the Lecidiplodia theobromii on the Costus speciosa, then Dendryphion penicillatum on Opium, then Diporthia, the Phomopsis fasciolorum, on Oroxylum indicum, Botrytis cinerea, on Vernonia and Thelmentica, then the perfect stage of the Fusarium graminearum, that is the Gibberella Z, and rice seed. You see, under the incubation method, I am trying here to show you how many pathogens can be detected during the incubation method. One of the most devastating pathogen, that is the Pyracularia. You can see over here how does it look under the compound microscopy, as well as under the stereo binocular one. The extreme top left side, that is the under the stereo binocular, where these two, whereas the third one is under the compound microscopy. The pyriform shape, the conidia can be very well visualized. Then the ear scab, that is Gibberella Z, and the seeds, you can see under the stereo binocular the perithecia under the that black colored beaked one. And if you tease them, the SI will come out. Under the compound microscopy, you can see the ascospores also. Apart from blast and Gibberella, the brown spot that caused by the bipolar psoriasis, you see the growth of the conidiophore and conidia, the attachment of the conidia and the conidiophores, as well as the conidia under the compound microscopy. And a very common fungus, that is the Phoma sorghina, that causes the gloom blight. You can see the pycnidia, how they are developed on the seed surface, okay, and on the blotter itself, and under the compound microscope, how the streaming of the conidia from the pycnidia is there. Okay, so these are some more pathogens again from the rice, like the Fusarium dimerum. You will be surprised to know this Fusarium dimerum is a very common human pathogenic, but we have been detecting a number of samples during our incubation method. And another important, that the stack burn caused by the Alternaria padwicki, you can see how the growth under the compound microscopy, the spores under the compound microscopy, as well as the growth under the stereobinocular. A very common pathogen Fusarium verticillitis caused by the Bacane, that causes Bacane disease, and you can see how does it look under the stereobinocular as well as under the compound microscopy. These are some of the pathogens of Brassica, related to Brassica, like the Leptosphaeria maculans. You can see that the pycnidia are produced on the seed surface, and from the pycnidia there is a purplish oozing, that is only the ooze conidia, which can be visualized over here under this picture. Another one, two species of the Alternaria, that is the Alternaria brassicicola and Alternaria brassici. Whenever Alternaria brassici is there, the conidia attached on the conidiophore. Whereas in Alternaria brassicicola, there is a chain of acropetal chain of the conidia. So these two species very important for Brassica. They cause leaf blight especially. Then the sclerotia of the Sclerotinia sclerosum, the admixture in the seeds sample. Apart from these two more, like Bipolis sorghicola, another minor pathogen of the millets, but nowadays it is common in millets, and we have detected in Brassica also. Okay. Now I'm just escaping. I've already covered somewhere. So whenever we stuck in identification part, through our routine or the conventional taxonomy under the CDL testing, we take the help of the PCR-based diagnostics. So Dr. Pradeep, our colleague, do the molecular identification and detection. Like we have confirmed the association of the Gymnosporium americanum and Diporthe fasciculorum in these two crops using the PCR-based sequencing. These are some more pathogens. Like under the incubation method, we could detect the presence of the Diplodia maydis, Diplodia microspora. These two are very important from maize. Okay, apart from maize, we have also detected the Diporthe fasciculorum as well as Botryoplodia theobromii from Jatropha. And I tell you about the Jatropha. When we imported first time, we could detect the association of these two pathogens. And apart from these Macrophomina fasciculina, very common seed-borne pathogen, and we have detected in mango stones, then the cucurbits, several species of the cucurbits, then the Pestalozia jupini. How does it look? You can see over here under the stereo, as well as then under the compound microscopy. And from tomato, we could detect the Phoma sorghina. How does it look? You can see under the compound as well as the stereobinocular. Apart from blotter, one more test, like grow out test for the same seedling symptom test we do sometimes when if it is required. Yeah. In under this, what we do, we grow the seeds and we try to observe the symptoms associated with, because some of the pathogens, they grow along the seedling. Symptoms in the seedling stage, like this over here, you can see under water agar, the blotter, then the potted. Okay. And very interesting, during the incubation test, many times we fail to get anything, because there are some laboratory saprophytes which are really menace for us. Only 2-3 examples I have shown over here, like the Melanospora zemi, you can see. Okay, and this Melanospora zemi is a typical laboratory saprophyte, and you can see the ascospore, how does it look, and how it get released from the perithecia. Okay, and this Melanospora zemi is so devastating saprophyte that the government of Australia quarantine department has declared this saprophyte as a quarantine fungus. So you can see that you can just realize the importance of the saprophyte also, apart from the seed-borne pathogens. These are some more saprophytes like Curvularia, different species of Curvularia, then the Cladosporium, then the Aspergillus. These are all very common saprophyte which we come across during our incubation method. So without the primary information and knowledge about the morphological structures associated with these pathogens, we cannot conduct our seed health testing using the blotter test very precisely. And apart from diagnostics like detection and identification, our duty is to manage, to treat. So our holistic approach is to do the disease survey, then the regulatory control, then the diagnostic protocol, availability of diagnostic protocols, then the salvaging treatments. So under these, there are different parameters like the seed certification, then the alteration in sowing and transplanting, then the field sanitation and isolation distance, then the field inspection, then the use of crop rotation, then the use of resistant varieties, then the detection and diagnosis, and finally, if we find something objectionable, then the destruction or the incineration. And this is how the disease-free harvest can be obtained. And what are the available treatment techniques which we usually adopt in our quarantine? That is the mechanical cleaning, the hot water treatment, and the alcohol wash, the seed dressing. So grow out. These are all we basically use for our seed-borne fungi and bacteria. You can see the cleaning applicable to both. Then the hot water treatment, it is again applicable to both. Then the alcohol wash, mainly for the fungi. And in case of rust, especially, we do the hot water alcohol wash. Then the seed dressing, especially for the fungal infection, the grow out for both fungi and bacteria. These are some of the tolerated levels of the seed-borne diseases in different crops. You can see like head smut, how many plants per hectare can be considered while doing the seed health, that seed certification, then the loose smut of maize, then the burnt, then the halo blight of beans. You can see over here the common blight, then the bacterial blight, the botrytis head rot in case of sunflower, then the sclerotinia wilt and head rot again. So you can see maximum 5 plants per 1,000 plant is a tolerated level. These are some of the levels, permissible limit of the seed-borne pathogens in case of certified and foundation seeds as per the Indian minimum seed standards. You can see for barley, oat, rye, wheat, sorghum, and bajra. So for categories of foundation and certified seeds, they have very clearly mentioned for different pathogens like Astellogonuda, it is 0.1% for foundation and 0.5% for certified seeds. Similarly for other diseases also. Okay. So these are some of the publications which we brought out using our conventional as well as the advanced molecular diagnostic techniques. And these are some of the useful links which we usually use for our detection and identification purpose. Okay. And these are some of the publications from our NBPGR, Division of NBP Plant Quarantine as a reference material. Okay. Now, as a routine, we have been organizing the capacity building programs and workshops to make awareness as well as the interest in the stakeholders, especially the scientists and those who are in the seed health testing industry. So our objective is to save our experimental stations, agricultural, horticultural feed, and also to reduce the crop losses. And last but not the least, I will say our aim is to create interest and awareness among the stakeholders through precision testing and treatment. And at last, I would like to acknowledge the director and PGR, head of the division, scientists, technical staff, and all those who are directly or indirectly involved in helping us. Thank you very much. Dr. Subhas.
Sir, now
Yeah.
Dr. Pradeep will take.
Yeah, sure.
Okay.
Unshare, sir.
Unshare, sir.
Just a minute, just a minute. Is it okay?
Yes, sir. I request Dr. Pradeep to share your slide, please.
Dr. Subhash, can I say something?
Yes, yes, madam.
This is Dr. Jamil has covered. One is quarantine and another one is seed health testing. For the benefit of the participants, I would like to clarify that in quarantine, there are no tolerance limits. Tolerance limit is 0. What he has shown MC as minimum seed certification standards, tolerance levels, that is for domestic material. I just want to clarify that.
Okay, thank you, madam.
Okay. Good morning. Am I audible?
Yes, yes, very much audible.
Slide also visible?
Yes,
Audible,
slide
sir.
visible.
Okay, thank you. So I will discuss about the molecular detection methods. Dr. Jamil sir already elaborated about the traditional methods. So if there is any problem, any issue with the traditional detection methods, then only we will go for the molecular methods. As we know that there are more than 1500 fungi, seed-borne fungi, and more than 300 bacterial pathogens. All are very difficult to identify by 1 or 2 classical taxonomists. So more or less somewhere we require molecular detection methods, either for their confirmation or for the validations. So I have limited time. I will try to show some videos if possible. Otherwise, I will explain. So first we will go for the lab visits.
This is plant
Is it
pathology
visible?
lab. All the work related to fungus and bacteria are carried out here.
Dr. Pradeep,
After
I think
quarantine
you
processing
separately
of
play
seeds which
because
are meant for exchange and seed
from
health
link
testing.
it will not, it's not showing.
Okay, okay. I will try directly.
Yeah, directly you play.
After quarantine processing of seeds which are meant for exchange and seed health testing.
No, you share first. Yes.
This is plant pathology lab. All the work related to fungus and bacteria are carried out here. After quarantine processing of seeds which are meant for exchange and seed health testing are plated on Petri plates containing blotter sheets. So plates are examined
Video is
after
not
8 days
showing,
of
Dr.
inoculation for the presence of fungal and bacterial pathogens.
It's not visible.
Not visible.
Okay, that's it.
Any other, I think if anybody else can visible in their screen, their participants.
No, doctor, no.
Okay, I will explain that.
Okay.
Okay, now visible.
Yes, it's visible.
Okay, so I will explain briefly, as I only 15 minutes. So as I tell, traditional methods are very important, but to assist them, we are doing molecular based detection methods. And there are 2 important steps or 2 important methods we are using for molecular based detections. We are using either conjured gene sequences based detection or species specific primer we are developed. These 2 methods we are using for the detections of the both fungi, seed-borne fungi, and the bacteria. So this is the outline of the practicals. First, we will do isolation of the fungal pathogen or bacterial pathogens, then genomic DNA extractions by CTAM method for fungi and bacteria. And there are specific conjured gene sequences like TAF, alpha, beta-tubulin, ITS, other genes which we are targeting for the amplifications for the fungus, and for the bacteria we are using 16S rRNA. And then we are doing sequencing, and based on the sequence identity matching with the NCBI BLAST, we are identifying the pathogens. Another one is we are designing species-specific markers from the sequence polymorphism. Either we are using the same sequence like TAP, beta conjured gene sequence, or any specific sequence for the development of markers. I will explain detail. So most important thing is the isolation of the fungus that sir explained. Then we are doing DNA extractions. Molecules start with the extraction of the DNA. These are the different reagents we are using for the DNA extraction, like EDTA 0.5 millimolar, tris buffer 1 molar, sodium chloride, sodium acetate, T buffer, and phenolchloroform isomer alcohol, isopropanols. And in this extraction, we need extraction buffer. That we use the above reagents and make a particular concentration, like CTAB we use 2%, EDTA, tris buffer, sodium chloride, beta mercaptadinol, and we add beta mercaptadinol after incubation period because it is degraded at high temperatures. And these are the equipment we are using: water bath, centrifuge, pipettes, pestle and mortar, and defridge. So these are the general steps for the DNA extraction. First, for the DNA extraction, I will explain of the fungus, and this is using by the CTAB method. That is a manual method. Kits are also available. I will explain CTAB methods. So first, we need pure cultures of the fungus. Then we are incubate in the BDB, product exposed growth for 7 days for the growth of the fungus. Then we do filtrations, and filtrate were harvested, and then this filtrate was used for the DNA extraction. First, we will grind with the liquid nitrogen, and fine grinding is very important, and grinding should be done either clockwise or anti-clockwise. If you will do grinding the both way, then there is a chance of degradation of the DNA. So this is important step for grinding. And then we add the extraction buffer that I have told in the earlier slide. We make the extraction buffer. Then we add 10 ml of pre-marm extraction buffer, and then we incubate at 65 degrees centigrade for nearly 1 hour. And this incubation, we also need to standardize time at least for the different fungus. Then we are adding equal volume of the phenolchloroform isomer alcohol, and then mix gently. Then we use centrifugation at 10,000 RPM for 20 minutes at the room temperatures. And then we separate the supernatant and the liquid supernatant from the tubes. And this step is repeated 2 times. And then we add 0.6 volume of the chilled isopropyl alcohol. It should be stored at -20 degrees centigrade or 4 degrees centigrade. And then we add 0.1 volume of the sodium acetate and incubate overnight. And then there is a formation of this like textures. These are the DNA. Then we again centrifuge on the next day, or after 1 or 2 hours, depending upon the protocol, we need to standardize. And then we wash the DNA with the 70% ethanol, air dry, and then we run the jar to check the quality and quantity of the DNA. As video is, we have video, but I don't think video is playing. That's why I'm not showing the video. Most important thing is that we should know there are many components that are used for the DNA extraction. We should know function, why we are using these different components like CTAB. This method is named by the name of the CTAB. So CTAB is used to disturb the membranes, and it forms the insoluble complex with the protein and most acid polysaccharides. That's why it leaving behind the nucleic acids. Then phenolchloroform isomer alcohol, it is used to separate organic contaminants into the organic phase. Then sodium hypochlorite, sodium chloride, it is used to help remove the protein and the polysaccharides. Beta mercaptadinol, it is used to prevent the polymerizations of the tannins. Then RNA is because when we extract the DNA, then both DNA and RNA will come. So we have to remove RNA so that it should not interfere in the further reactions. So we use RNase A to remove the RNA. Then isopropanol is used to precipitate the DNA so that other impurity can be removed. That sodium acetate, it is basically used to neutralize the negative charge of the nucleic acids. Then EDTA, it's deactivate the enzymes and by reducing the activity of the DNA or RNAs. Because DNA is there, that will degrade the DNA. And tris buffer, it is used as a buffering agent so that DNA will remain stable in the buffer. So after that DNA extraction, we need to check either this DNA is pure or not, it is good quality or not, because it will affect the further subsequent PCR reactions. So most commonly we use nanodrop for the quantification of the DNA, and this is simple formula. Amount of the DNA is we use the OD at 260 into 50 into dilution factor by 1000. So it is the concentration of DNA in the nanogram per microliter, and for the quality we need to take 260 by 280 ratio, absorbance at 260 and 280, and for the DNA it should be around 1.5. More or less may have contamination of either the RNA or protein or phenols. So quantification very important. Other way we are also doing quantification by using the gel. 0.8% gel is used for the quantification of the DNA, and for that gel quantification we use lambda standards. Their standard are available for different concentration like 100 nanogram, 50 nanogram, 1000 nanograms. So we are comparing with this, and roughly we can estimate the concentration of the DNA by using the lambda standards. So quality is very important. If we run the gel 0.8%, if we get this type of the gel, this means it is not good quality. This contamination at lower size, this is mainly due to the RNA, and this should not be the DNA. Pure DNA should be intact like this. There should be no lower band RNA or maybe phenol. This may be due to majorly due to RNA, and band should be intact. So this is a good quality DNA, so that we can use this for the PCR reactions. If there is a contamination, either phenol or RNA, that will interfere in your PCR reactions. So these are the different conserved genes we are commonly using: ITS, COX gene, beta-tubulin genes, TAF gene, large subunit and small subunit for the fungus. And for the bacteria, we are mostly using 16S rRNA. These are conserved genes. So we have developed different markers by using different genes. Like ITS, we have developed bipolaris medis. For large subunit, we have developed marker for the micropomina, flagellina, TAF, beta-tubulin. So we have developed different markers using different genes, or we can also detect based on these genes, or we can use multiple. If we are unable to identify using a single genes, so we can use multiple genes for confirmation and validations. So this is the PCR. The next step is the PCR. So PCR is the polymerase chain reactions, in which we can amplify single copy into the multiple copies, 2n copies. So it has 3 steps: denaturation, annealing, and extensions. This is explained in this figure. There is a first, there is a denaturation at the high temperatures, 94 or 95 degrees centigrade. Then there is a primer binding. Second step is the primer binding, and then amplifications. So for this, this process is similar to the in vivo that is happening in the any person, living beings. So this is mimic of that PCR. Here we use all the artificial conditions. So these are the different components we are using. DNTPs we are required because we need to amplify the DNA. So NTPs will bind to the amplifications. Primer are required to start the amplifications. Tag DNA polymerase, it is enzymes that amplify the DNA. Attach different DNTPs will be attached with the help of the tag DNA polymerase. Then magnesium chloride, it is used for the standardizations and particular amplification. At if this concentration is very high, then non-specific amplification will occur. If low, that there will be no amplifications. And tag buffer, it will give the buffering to the reaction conditions. And these are the different steps like denaturations. There are 2 steps. Initial denaturation means we have to denature all the DNA that is available with the PCRs. Then each step there is a denaturations. Then primer annealing, binding of the primer at it is also occur at specific temperatures. Then primer extensor generally 72 degrees centigrade, but time may be vary. If there is 1 minute, in average, approximate 1000 base pair will be extended in 1 minute. So it may be less, more or less based on the product size. And then there is final extension, mean total product, if anything remain and extended, that can be extended in the final steps. So most critical step is the designing of the primer in the PCRs. So primer designing is a very critical step. For this specific band obtained from the amplification with the target pathogen will be sequenced. Then sequence were aligned within the bioedit tools, and alignment of the nucleotide sequence with different pathogens. We extract the sequences from the all related pathogen from the NCBI database, and then we are comparing with that one. And then unique regions of that particular sequence we are designing, we are try to identify that region and designing the primer from that part, so that it should not amplify in the other pathogen, all the related pathogens. So critical is the primer length. It should be 18 to 28 base pairs. Then primer should be unique, should not amplify other related pathogens, other different species of the same genus or pathogen of the same plants. And then GC content should be optimally 50 degrees centigrade. If it is high, then annealing temperature will increase, and that will interfere in the extension steps. and there should not be any cross reactivity. Primer dimer formation should be avoided. If there is a primer dimer formation, then they will bind themselves and they will not bind to the template. Then self compatibility should be avoided, and melting temperature compatible. So like this, these are the very similar sequence. We are searching for the variability in the sequences. If there is 2, 3, 5, or at least 5 or 10 base pair differences, then we try to target that region and designing of the both forward and the reverse primers in that regions. So other step is the specificity. So specificity and sensitivity, two steps are very important for the any detection protocol we are developed. Specificity means it should be amplify only targeted pathogen, not in the other pathogens. So we are confirming the specificity by using the all other related pathogens. For example, if we are designing pathogen for the Diporthi longicola, then we are taking all is closely relative Diporthi other species and the pathogen of that particular crop, and it should not amplify in the other. Like this, it is only amplified in the Diporthi longicola, not in other species. These all species that are related to these pathogens. Specificity and then sensitivity, up to how much level we able to detect that pathogen, that is sensitivity. Either we able to detect in the 100 nanogram, 10 nanogram, 0.1 nanogram. So these two, sensitivity and specificity, are the most important for any sensitive detection protocols. So these are the different steps which are involved in the sensitivity. So along with the PCR, we are always using real-time PCR based detection methods. In real-time PCR, difference is that we need not to do gel electrophoresis, because we don't need that one, and it is time saving and more sensitive than the PCR based methods, classical PCR based methods. So it's allow detection of the low copy target with greater confidence. It has a very wide range of copies detectable within single runs, and if fluorescence increase, that is directly proportional to the amount of template, and it save time, as already explained. Then if we have developed any detection protocol, then we need to use that protocols. So we are using, our major aim is to detect directly from the seed. If there is a seed is infected, either we can able to detect directly without incubation, because incubation will take at least 7 to 8 days. So we want to save the time. So we use directly infected seed. We are extracting DNA, and we are checking either our protocol is efficient or not, detecting from the directly seed. If we able to see that we can detect the particular pathogen in 4 to 5 hours instead of incubation period of 7 to 8 days by classical methods. So our detection protocol we use, we extract the DNA directly from the infected seeds, and then total seed we use in that DNA, both plant DNA and that particular pathogen DNA also, and then we run pathogen specific markers, and we able to detect that using the conventional and both qPCRs, that is real-time PCRs. So our major aim is to detect directly from the infected seeds. So other than this, we are also doing multiplexing. Multiplexing mean we are detecting more than one pathogen in the single run. So either it is duplex, triplex, or tetraplex. So in that, if we suppose any pathogen infected the more than any seed infected more than one pathogens, either we able to detect in the single run. So for that, we developed the multiplex PCR based protocol. Like in this, we have developed for the 3 pathogens. This is individually. First, we need to develop individual protocols, and then we designed in such a way that we able to detect all in a single load. Like there should be a difference in the base pair, at least 75 to 100 base pair difference. So these are the single, and this is multiplex. All 3 pathogen is a single tube we will able to detect. This is a little bit more challenging. We need to design the primer very carefully. They should not be bind with other primers. Their length should be unique. Their handling temperature should be nearly same. So there are different challenges during the development of multiplex PCR. But these again save the times. If we able to detect the single PCR in 4 or 5 hours, same way 4 or 5 pathogen we can detect in same time. So it will save time and resources also. With the same set of the chemicals, we can detect 3 pathogen in a single lot. So other than this, we are also doing phylogenetic analysis and genetic diversity analysis mainly to check either we are receiving pathogen from the outside India. Is there any variability from the Indian pathogens and the abroad pathogen? If there is more variability, then that may be more virulent or more pathogenic. That why we are regularly doing these studies also. How much variability is there in the Indian pathogens and outside isolates. And also there is some anomaly in the many groups, like this study we have used the phylogenetic analysis of bipolis, Carularian, Exurillum. There are many times there is change the nomenclature. Many species of the bipolaris went in the Carularia and Exurillum and one way or other way. That's why we are using this phylogenetic analysis to really identify which group this particular species belong to. So in some way, I think I have finished my time. Molecular technique play a key role for the detection and identification of the several fungal pathogens of the quarantine significance from the seed and planting materials. And the molecular technique advantage, and they are quick, accurate, efficient, and over the morphological techniques. Molecular technique are also more, very important because we cannot detect each and every pathogen by molecular technique, because in quarantine we are receiving lakhs of samples. So molecular technique are most important, but molecular technique are as is over that for quick detections. And for safe exchange of the germplasm, the conventional seed testing method in combination with the molecular diagnostic play a key role in the minimizing the risk. So there are many laboratory safety protocols we need to follow. And I want to thank you from the directors, organizers, our plant pathology lab, and special thanks to Rajkan and Radhika for the videos, but I'm not able to show the video. Sorry for that. And thank you. Thank you.
Thank you, Dr. Pradeep. Now I request our participants, if you have any question, you can ask. Or you can also post any question if you have. There is one question, sir. What are the minimum seed requirement for embryo count method?
Subhas, as per standard procedure, 1,000 seed is required.
1,000 seed, okay.
Yeah.
Any other question? Participants, you have any question, you can ask, or you can send in chat box. I think there are no question. Nobody is like to ask. Anybody? Okay. Thank you, sir. And now, if there are no question, then I may propose a vote of thanks. I, on my behalf and on behalf of organizers, I thanks to Dr. Jamil Akhtar and Dr. Pradeep for showing practical on plant quarantine processing in respect of fungi and bacteria detection. Sir, you both beautifully explained pathogen detection using various methods, as such as visual examination, grow out test, compound microscope, and stereo microscope test, and also through molecular techniques such as a PCR detection. And you also show some of them through videography. Sir, both of you are thank you very much again, and thank you very much.
Thank you, Dr. Thank you.
Thank you, Dr. Subhas. So can we leave now?
Yes, sir. Now I request all the participants to join at 12. Sharp and 12. Because we have interactive section with our stalwart, Dr. Paroda. Okay, thank you very much. Thank you for patience listening to the speakers.
Director, sir, esteemed HODs, OICs, organizing team members, and participants in this training program interaction session. So first of all, I would like to request our director to welcome Dr. R.S. Parada sir with the bouquet. Chief guest of today's function. Thank you very much, sir. Now, I would like to call upon Dr. R.K. Gautam, Head Division of General Plan Evaluation, for kind introduction and welcome address, please, sir.
Thank you, Dr. Jyoti. Namaskar. Good afternoon. It is really an auspicious day, and we are very lucky to have the gracious presence of the most revered and our chief guest, Padam Bhushan, Dr. R.S. Baroda, former Director General ICAR and Secretary Dear Government of India. Sir, on behalf of the entire NVPGR family, we profusely welcome you, and we all feel highly inspired and elated by your kind presence in your own campus of ICAR and NVC. We are also blessed with the able guidance, support, and dynamic leadership of our director, NVPGR, Dr. G.P. Singh, who is also our training director. Sir, we also welcome you in today's program. Sir, as we know, ICAR NVPGR is nationally and internationally acclaimed for its pivotal role in PGR management. Considering this, Council directed the NVPGR to organize a 21 days training program on PGR management. Accordingly, we conceptualized and designed this training program with the valuable guidance and support of our director. The training is financially supported by Human Resource Management Unit of ICAR. We especially added the component of utilization in this training because the PGRs become valuable only when these are properly characterized, evaluated, and utilized in proper programs. So in this training, sir, we additionally put the component of evaluation, germplasm characterization, and utilization part in more amounts. Sir. The resource persons are experts from NVPGR, other institutes in India, and many from CG centers. The training is planned for the early career scientists and faculty members so that the training can go a long way in enriching our young faculty members. Therefore, 84 trainees representing 19 states and 39 institutes and universities constitute the diversity of the participation. Besides government organizations, trainees are from private sector and city center also. So today we have kept this interactive session to inspire and benefit our all trainees with your wisdom and guidance. But before that, it is important to introduce our chief guest to all the trainees. Dr. Raj Paroda, former Director General, Indian Council of Agriculture Research and Secretary, Department of Agriculture Research and Education, Government of India, is an accomplished plant breeder, a visionary research administrator, development practitioner, and highly sought after policy advisor. Dr. Paroda ably transformed the Indian National Agricultural Research, Education, and Extension System while serving as Secretary, Department of Agriculture Research and Education, Ministry of Agriculture, Government of India, and Director General of the ICAR during a period of 7 years. This vision led to creation of more than 30 new institutions in different sectors of agriculture, like crops, horticulture, livestock, natural resource management, fishery, agricultural engineering, and social science. This strengthened the NARE as one of the leading research and development organizations in the world. India's National Gene Bank at New Delhi is the second largest in the world, which is having about 4.7 lakh germplasm accessions across crops. Dr. Parodaji could seek a grant from USAID to build the entire new complex of NVPGR while serving as its director from 1985 to 87. The Gene Bank was inaugurated by the then Vice President of India, Dr. K.R. Narayananji. Besides building the most modern gene bank at the National Bureau of Plant Genetic Resources, he created 4 bureaus on genetic resources of animals, fish, microbes, and useful insects. As a result, India today has the best network of institutions to manage its valuable genetic resources. His deep passion for genetic resource management led him to make special efforts in establishing gene banks in all 8 countries in Central Asia and Caucasus region, while heading the CGIR program for CAC Tashkent. In recognition of his exceptional contributions in the field of genetic resource management, the International Crop Research Institute for Semi-Arid Tropics, ICRISAT, and the Agriculture Research Institute of Kazakhstan have named their gene banks after Dr. Paroda. In fact, he is nicknamed as Gene Bank Guru by his friends and admirers. It was his foresight and determination that led to the construction of the most unique National Agricultural Science Center, NASC, the most pride place for all of us. The NAS complex was built in record time of 5 years and inaugurated in 2001 by Honorable Prime Minister Shri Atal Bihari Vajpayee. Dr. Paroda was instrumental to establish the Indian Society of Plant Genetic Resources at NBPGR in 1987. Since then, ISPGR, the society, has undertaken several activities under his visionary leadership. Special mention is of the first International Agrobiodiversity Congress held in 2016, which was inaugurated by Prime Minister Shri Narendra Modi ji. His lifetime work in the service of national agriculture, especially for the popularization of science and to link it to society's exemplary, and has been duly recognized in the form of several prestigious national and international awards, including Padma Bhushan, conferred by Government of India in 1998. He is recipient of several awards and recognitions, namely Rafi Ahmad Kidwai Prize, ICR Team Research Award, FICCI Award, Om Prakash Bhasin Award, BP Pal Gold Medal, Norman E. Barlow Award, Agriculture Today Award, Mahendra Shiromani Award, A.B. Joshi Award, US Avasti IFCO Award, and MS Swaminathan Award. He has also served as President Indian Science Congress and President National Academy of Agricultural Sciences. 19 universities have awarded him DSc degree, including Ohio State University of the US and Indian Agriculture Research Institute. He is fellow of almost all science academies in India, Third World Academy of Sciences, Italy, Russian Academy of Agricultural Sciences, Agriculture Academics of Armenia, Georgia, and Tajikistan, and honorary member of American Society of Crop Science and American Society of Agronomy. Recently, he has been elected as Foreign Fellow of Australian Academy of Technological Sciences and Engineering. Dr. Paroda had been the founder chairman of Global Forum on Agriculture Research from 1997 to 2002, based at Rome. He also served for more than 2 decades as Executive Secretary of Asia Pacific Association of Agriculture Research Institutions, popularly called as APARI, which is based in FU, Bangkok. Dr. Paroda also served as Chairman Farmers Commission of Haryana from 2010 to 2014. He was also a member of Advisory Committee of ACIR, KB, and WMO. He was Chairman of Board of Trustees of ICRISAT and member of Finance Committee and CMEC of CGIR. Currently, he is serving on the board of International Fertilizer Development Center, Alabama, USA. For the last over 2 decades, Dr. Paroda is serving as founder, chairman, Trust for Advancement of Agricultural Sciences, popularly called as TAS, which is based at Indian Agriculture Research Institute, New Delhi. So I request all the attendees to unmute themselves, and I request all here to kindly give a big round of applause to our chief guest. Humble introduction. I once again welcome our chief guest, sir. I also welcome one and all present on this occasion. Thank you. Thank you. Namaskar.
Thank you very much, sir, for making us aware about enormous contribution of Dr. R.S. Paroda, sir. So now, as 2 weeks of the training programs have been passed, I would like to brief about the training program, please, sir, with your permission. So, sir, we welcome you to this session of the training program. So, this 21 days training program about the utilization and management of plant genetic resources being conducted from 1st February onwards. It is jointly organized by ICR and BPGR and HRM unit of ICAR. So in this training program, sir, we have received more than 120 applications. Out of that, we selected 84 participants, depending upon the Zoom slots, limited Zoom slots. These participants represent different states, 19 states they are represented, then different various institutes. So among the students, Madhya Pradesh and Odisha is having maximum of 12 participants from each. Then among the institutes from SAUs, 59 candidates are there, from ICR 9, CG Institute 2 are there, private 6, CAU 1 and others. So we have good representation of gender as 33 of 84 are female participants here. And regarding designation wise, most of them are assistant professor, 65 are assistant professor, 12 are associate professor or senior scientist, 5 are professor and principal scientist, and some also 2 are technical officer. So now these are view of participants. Now coming to this training program was inaugurated on 1st February by Deputy Director General Education Dr. R.C. Agarwal and ADG Dr. D.K. Yadav sir. In this training program, sir, we have several resource persons, 81 resource persons we have selected. We wanted to cover all the area of PGR management and utilization. In addition, we have also selected topics like phenomics, then big data analysis, statistical analysis, nanotechnology, AI in agriculture, those things. So among these, 7 speakers were international speakers belonging to ICARDA, CIMMIT, Kansas State University, BISA, World Vegetable Center, and among the external speakers, 24. Those were from ICR institutes, the State Agriculture University, Botanical Survey of India, and some other CSIR institutes. Now these are some glimpses of the speakers, like Dr. S.D. Upadhyay from ICRISAT, he talked about core and mini-core collection. Dr. A.K. Joshi from CIMMIT, he talked about conservation, exchange, and use of wheat genetic resources in the global context. His talk was very much appreciated, sir. Then we had some novel topics like trans situ conservation of plant genetic resources by Dr. Anuradha Agarwal, who was the former colleague at MBPGR. Then we had biosecurity issue talked by Dr. S.C. Dubey, ADG plant protection. Then we had the topics by Dr. Martin Van from the World Vegetable Center. He talked about the vegetable genetic resource utilization and all. Then PGR informatics. The Sunil Achep is the man of PGR informatics in the Bureau and in India also. He talked about enhanced access to information for enhanced utilization. Furthermore, we had talk from Navtej Bens from PU Ludhiana. He talked about breeding and genetic enhancement in cereal crops. So he covered rice as well as wheat. Okay. Then some talk from R.K. Singh. Then millet, we covered International Year of Millet is being organized. So Dr. Tara Satpati. Then we had talked from Dr. Shiv Kumar Agarwal from ICARDA. He talked about genetic gain in the pulses. So with this, I have briefed about the training program. So introduction program.
Before we begin with the interaction with the colleague, because as informed by he has limited time. So let us hear him. I would request you to kindly address for some time, and then we can interact and give your blessings to the young colleagues who have joined online and also offline. So I take this honor to invite you to address the colleagues who are there as a participant and also otherwise as a faculty member. Thank you, sir.
Thank you, Dr. Singh, Dr. Gautam, Dr. Jyoti Kumari, Dr. Huda, all other members of NDPGR, here participants of this important training course, which I consider very relevant in today's context, because it is addressing the management and utilization of plant genetic resources And I'm happy to interact with you. Also, the generous introduction provided by Gautam. I would like to say thank you very much. It's so kind of you. But all what could be achieved was because of serving the society with human face. And I'm happy that our agriculture research system has grown stronger. And today it is making its impact globally considered as one of the strong agriculture research systems. And our economy is also growing. We have become one of the 7 largest exporters of agriculture produce in the whole world. And we are aiming for 5 trillion economy. From 2.5, we have already just now 3.5, leaving behind many European countries. And hopefully we would be among the first 5 strong economies in the whole world. Having said this, I was associated with the Green Revolution as a postgraduate student at IRI. At that time, the seeds of Green Revolution were being sown. I know how important the genetic resources are. If we had not utilized those dwarfing genes from rice and wheat, Norintane and DGOgen, possibly all that revolution would have not happened. So I don't have to tell you how important genetic resources are. And we also have to remember that this is a subject in which you have dual role. One is to guide yourself through scientific contribution and research. And the second is you have to serve the society. Serving the society becomes very important and should not be neglected while we work in the area of plant genetic resources. And Green Revolution, when I discussed with Dr. Baldog, what do you think are the cradles of Green Revolution? And why it is not happening in Africa after it had been achieved in India? So he was working there, the Sasagama Foundation, and with all donor support, after Green Revolution happened in South Asia, almost after 10 years or so, his reply was that I cannot get the same policy support which we got here in India. First cradle of success is policy support. Second was that from where do I get institution like IRI in Africa? So the institutional support. And third one was that from where do I get human resource, the kind of Swaminathan and the farmers of Haryana and Punjab who are willing to spend once they are aware about the importance of any particular technology. The fourth one was the partnership. Partnership not at only national but at international level. If we didn't have partnership with Simit and Erie, possibly that would have not happened. If we didn't have institutions like IRI, where I also studied, and the human resource was not developed, possibly we would have not done that. So I recognize, and luckily, having studied there, having seen Green Revolution taking place, having gone to greener pasture, for postdoctoral research and getting offers which often people get to remain outside. I took a challenge to come back. And I thought that these cadres are important. Institutions we must build. Luckily, I had the opportunity to serve NBPGR. And my goal was that maybe tomorrow, if not today, it will also be rated as one of the best institutions like IRI. And I must compliment and congratulate you because other than the national institutes, the rating was done by ICR last year and NBPGR got second ranking. This would have not been possible had there not been institution and human resource. But when I joined, genetic resource activities were considered only as a service function and not as science. We had to start the postgraduate program also, which I wanted as director here, NBPGR was never successful because the academic council of IRA would not approve it. And I became DDG, I again tried, but again it would not be successful. When I became director general, I said first thing is the course has to start. The question is, I'm not saying that I did it, but I recognize that you need it. You need institution. When I was working for my postdoc and coming back, where in fact even Dr. H.K. Jain had studied for his PhD with the same expert with whom I was associated, Professor Hugh Rees, FRS. And Jain also had come there to attend a conference in Oxford. He asked both of us a question that Jain and Raj, why I have many students from India go back, but I don't hear anything except a few like you and what I see this young man, but I don't know whether he will be able to go and also be successful like you. So the message was why we are not able to perform in our environment. And when you discuss with your colleagues and friends, the answer was that you don't have that environment. You don't have people to support young people. The leaders want to take credit for themselves. And there is no teamwork. Mostly people are individualistic. So I thought that who is going to provide environment on silver plate? You are not that lucky born to get everything, which I think now young generation and many young scientists today are getting and seeing and working in these institutions. This environment, I don't think we had this environment. But then you have to create it. And when you create and when we created this institution also, it was not only the brick and mortar. The focus was that every scientist must get trained and exposed to the best conditions outside India. I don't think in ICR you find any institution where 100% of its scientists were trained abroad. Under a project, because I knew government will not allow. So we kept a provision in the project itself. So one could not. And then give the responsibility to withdraw as an agency to see that they go and they come back and go to the best institution. So the question is in brief that we require institution, we require human resource, and we require policy support. And policy support, who will give? Unless baby cries, your mother would not give milk. So the scientists have themselves a role as a leader, as a director, as deputy director general, to make sure what they need, they get. And that is our major challenge. So we feel that we have greater responsibility. So having given this background, I come back to genetic resource management and utilization. Firstly, India is very rich in genetic resources, one of the mega centers. Vavillovian Center. We had our own Vavillo, Dr. Harbhajan Singh. But imagine in those days when travel was so difficult, communications were difficult, visit and interactions were difficult. How could he go to all parts of the world and collect? So I went first as a director in NVPJ to go and see the Vavilov Institute and the gene bank which he built. Because if you don't see and then you don't act, then I think you are not doing justice. But seeing is always believing. So I must say that Dr. Harbhajan Singh work has to be very thoroughly documented. Dr. G.P. Singh, who can assign some consultancy agency or some retired people. Well, we don't have people like R.K. Arora and Chandel, but there must be still people who could be requested to document what actually, where he went, what did he do, how he attended, what he collected, and what were the difficulties. And I think there were two reports of those times. I have been emphasizing that those two reports must be digitized. So the first thing is what you have. Have you collected everything? We say we are the richest, one of the richest genetic centers in the world. But so when the NATP was negotiated, I requested Dr. Gautam, that time Director I NBPGR, to prepare a mission project for collecting genetic resources. Because that time we had around, I think, 200,000 accessions. I said, we will continue collecting another 50 years and then this 200,000 have come. Maybe another 100 years, another 200,000 will come. I said in 5 years' time, take it as mission mode and collect whatever best can be in partnership, not yourself alone, with other institutions. NBPGR has to also be a big brother and build partnership and do it even with the ICRI set also, whatever germ plan they had. I said that germ plan must be evaluated for those crops in our Indian conditions. So I provided multi-locations. We brought out catalogs. Those catalogs were printed. They must be here somewhere. And if ICRISAT has recognized whatever for the gene bank, it is not just because I was Director General. We worked together as partners and identified many useful materials and provided them support for exchange of genetic resources, created for them the facility at Hyderabad. That center was not existing. So the facility was created for the best quarantine system to be built and provided them all kinds of opportunity. So one is in the management part, I will come to utilization later. You have to see what you have with you. And what you have with you is not important. It must be known to the entire system what you have. It's not your personal locker in the bank. Whatever money you have, whether lakhs or crores, which you don't tell. Because here, if you don't tell, who will appreciate and realize that you have something? Firstly. And unless they know you have something, who will come to you? They will not come to you. ठीक है रखा होगा black box में बहुत कुछ पड़ा है. So gene bank should not remain black boxes. One point which I would like to say. And whatever we have, maybe still there are, duplicates. Lot of materials may not even regenerate. I'm not sure how frequently we have been rejuvenating them. And so we need a critical analysis of our gene bank. And what I was telling under that NATP mission, in 5 years' time, we collected another 200,000. I was not really sure that this will happen. 50 years, 200,000, 5 years' time, 200,000. आपके पास में जो आज है 400 और 500 की बात करते हैं, तो ये ऐसे ही नहीं हो गया. It required effort and a system to work. Now that kind of, system must be there, and you can't do it alone. You can't give the credit to me alone. अगर आप लोग काम नहीं किए होते, सारे लोग मिलके गए नहीं होते, मिशन को चलाया नहीं होता, पैसा देने से क्या होता है? बहुत पैसा देते हैं, तो काम होने वाली बात है ना. So what I intend to say is that whatever we try to see, first we should find out what is the need of the system, and for that need, what is required to achieve. Like, Abdul Kalam used to say, for any success you must have a dream. पहला तो ड्रीम तो हो, और ड्रीम को अचीव करने के लिए then what is required to be done? You need to have that kind of perseverance and sustained effort. Without that will not happen. तो वो करने के लिए फिर हमें, but once we know कि हमें वहां जाना है, वो हमारा टारगेट है, तो कोई मुश्किल नहीं है, मैं समझता हूँ. We Indians have shown, only problem is we perform under pressure, under stress, under calamity. We become complacent once we have enough. So that complacency must be guarded against. And in that respect, now, first thing is, whatever you have with you, वो अपनी जो आपके पास में है, चाहे वो कंकर है, चाहे वो फल है, उनको तो आप जान लो. उसके बाद में what you need, which you don't have, to get and from where to get, is an issue of exchange. So in the management side, then the exchange becomes very important. And many lectures would have told you, I'm not going into all those details. You can have a look at the PowerPoint presentation. I have that PowerPoint presentation of mine, which was there in the recent symposium.
We'll share that.
So share that with them. Share also that recent special issue of the genetic resources journal. And share also with them the proceedings of the first International Agrobiodiversity Congress and souvenir and Delhi Declaration. आप लोग भाई पढ़िएगा, थोड़ा time, homework should also be there. Course doesn't mean just attended classes and then finish. I was for a training course for 10 days at Oxford for the management with the CGIR leaders. And I can tell you, वो क्लास तो 1 घंटे की होती थी, पर उसके लिए पढ़ना कमरे में कम से कम 12 घंटे। Otherwise, you will be identified immediately that you are not studying and you are not aware and you are not prepared. So these trainings are very important. They should be taken seriously. There are resources which have been spent and you are fortunate to be participant in this. I see about 75 are there, I think. Or currently, how much are on the line?
75.
I see from here, I think, maybe. So I'm happy that out of 81, maybe some are absentees today.
Maybe sir, connection issue sometimes.
Connection problems also could be, yes. But it's a good number and this is again the benefit of COVID. We got that while sitting here, while saving resources, we can interact and exchange. And this is another, so in the crisis, always there are positive sides as well, which we have to understand. Positive side of COVID is that you have to now lay greater emphasis on local food systems, the food systems which are good for your health and immunity, not only for your food. So the UN conference was held recently, and it was the conference on local food systems. And we also had organized our conference at that time, when second IAC was organized, that we must have also a conference on local food system. And NBPGR has a niche for it. I was, while coming, also talking to your young scientist who came to pick me up. And I was telling that no one is going to work on rice bean, say moat bean, people were talking about buckwheat. They were talking of, the world over, people were talking of quinoa, and we have our own materials which have to be also taken care of. So there are those materials which people have forgotten, and we must identify and we must therefore also strengthen not only evaluation but your genetic improvement program and laboratory for genome editing and breeding, genomics and so on, because no one else is going to work, and that's how NBPGR name is there. No one was working on Bindi at that time, and Pusa Savni was the first variety, came out from Dr. Harbhajan Singh's work. And of course, IRA took the credit because IRA was the institute. But that is the work on genetic resource evaluation. And based on that, the MICO company came up. The seed company, today country is one of the biggest seed company. Basis was only that material. So you have to understand that how important we are. So coming then in the context of exchange, till today we are importing. And we would require to import. Our food basket would have not been different if it had not been exchanged. And when you look at, which we reviewed last time, countries like Australia, USA, they are 100% dependent on everything what they are today consuming and producing. They were never originated there. Origin of the plant and biodiversity took place around the civilization sites. Wherever the civilization took place, wherever ladies settled down, asked their nomad people to not to run around and just hunt and eat, but grow plants and then survive. That was the level where the whole genetic resource activity and human civilization started, whether it is Mohenjo-daro, whether it is Nile Valley, whether it is Yellow Valley, whether it is Mexican well. So we see that these genetic resources are important, not for us alone, but for tomorrow. If kiwi fruit we would have not imported when I was director of NBPGR, today it would have not become such an important fruit here. And we didn't know what will be its future, where we will grow it. How could you be predicted unless you try? And I go to Philippines, I go to Thailand. Thailand rambutan is there, such a good fruit there, and longan there. They are still not there here. Why they are not here? Now you get that kg guava, which is coming. So they have that guava which is not very sweet. Like our mango is very sweet. But Thailand and Philippine mango is not sweet. And that's why world over people like them. Whereas we have the maximum variability of mango. So it is again the issue of not only having something as very rich, but then trying to find out which will be more useful for the population, for the society, and what characteristics are required, and evaluation for those characteristics. So these are the issues which are important. Then coming the question of utilization of the genetic resources, I have already told you that we need to utilize them. For utilization, it is not important that you have a catalog indicating how many days for maturity and what is its height and what is the duration of the crop. Today we want, if I want fortified variety, then variation for zinc. If it is wheat, variation for gluten, variation for protein. Now millets we are talking, they have more fiber, they have more fat, they have more protein even than wheat and rice. So evaluation for those traits. I went to the gene bank of Korea, South Korea, and was looking at the research work they were doing. They were working on rice. So I said, okay, are you working on disease resistance or drought resistance? Or they said, no, nothing of that kind. We are working on medicinal value of rice seed. I said, rice seed? They said, yes, there is a lot of medicinal value in rice oil. We would, which means we are 50 years behind. If you think in that context. So the evaluation part, again, cannot be done individually. But NBPGR or those who are working with genetic resources at any place are the facilitators. They can facilitate the process and bring people together. And therefore, teamwork, interdisciplinary work, interinstitutional collaboration and work and partnership becomes very important for the evaluation part. And that evaluation we must do for those traits which are important and which we need, not for today, but for future, but for our future generation. And we need to see what we require still to be imported and import from where. I was in Uzbekistan. In the market, they are selling the almond, which is with shell, but you just press it and it breaks and you take out, and they are roasted, and also the roasted almond with shell is sold in the market. Is that not useful variability? But we don't have. But unless you know, unless you are aware who has what and what can we get. Now tomorrow we want to make rice wheat system to be diversified in North India. I see the possibility. People say maize can replace rice, but then again it will be serial rotation. It will not be sustainable rotation. I see the possibility of soybean and wheat. But soybean, we have very limited germ plant. And so soybean germ plant, whatever we have utilized it in the rain-fed area. We want now in irrigated material which can give you more than 3 to 4 tons of productivity. Then it will replace rice. So is there no need for more germ plant of soybean today in India? Are we having partnership with IITA? Are we having partnership with USAID? USAID has been our largest supporter. Let me tell you this. And in the project I got, I've also got sanction from Japan, JICA support for building the gene bank, which they had built in Philippines, also in Thailand, and in Sri Lanka. But I prefer to go for USAID because firstly that was limited, only 10 million they were giving, not more than 10 million. And they were saying that we will do it everything ourselves, turnkey basis, we will construct our own engineers, our scientists will come and they will work, they will train you. I said, they will come and train us, but my scientists will not go out and see what world is about, which I have seen. So USAID I approached and I told them that you owe me. You don't fund me, you owe me. And this is what actually Nile Brady was the Deputy Secretary in Agriculture, who was Director General of ERI. Luckily he understood. He knew importance of NBPGR, genetic resources, and being the Director General himself as a scientist. And then Dr. Atwal was at that time in Windrock. He was also a geneticist and a breeder. And in Windrock, he helped me मीटिंग कराने में आ जाओ तुम्हारे मिलाते हैं इससे बात करते हैं कई राउंड लगाए हमने 2 साल में बहुत पापड़ बेले थे पर फोकस यही रहा और एंड में मैंने उनको कह दिया डॉक्टर ब्राडी नाउ माय पेशेंस इज लॉस्ट we have worked we are coming to you maximum exchanges if you see between 2 countries or genetic resources, it is between USA and India. This analysis I had done. और वो NBPGR और USDA के थ्रू थी तो हमने कहा तुमने मेरे से इतना मटेरियल लिया है तुम मुझे दो वापस वो कहता है हमारी जीन बैंक भी इतनी इम्पोर्टेंट नहीं है जितनी बड़ी आप बना रहे हो और ये कर रहे हो मैंने कहा तो आप भी बना लेना मुझे तो बनाने दो and actually on that argument they came forward. So the point is that we have to have first the knowledge worldwide. We must act locally, but not to see only world and not to act at all by coming back. Secondly, we must see what we need. We needed, I joined as director, all heads of divisions had the air conditioned rooms. बहुत पुरानी सी बिल्डिंग थी वो उसी में हम भी बैठे मेरे कमरे में भी एक एयर कंडीशन था और जिसके पास जाऊँ मैंने कहा यार सीट कहाँ रखते हो तो जाके देखा तो सीट के लिए कमरे में एम्बियंस टेम्परेचर पे बोरियों में पड़ा था एक कमरे में मैंने कहा ये हमारी जीन बैंक है तो द क्वेश्चन इज दैट मैंने कहा भाई अब और कुछ हुआ ना नेक्स्ट जो रेफ्रिजरेटर या जो एयर कंडीशनर खरीदा जाएगा वो स्टील बैंक जहाँ आपने सीट रखा हुआ वहीं से शुरुआत करते हैं हालांकि दैट इज नॉट आइडियल वन बट कम से कम टेम्परेचर को थोड़ा तो कम करेगा द पॉइंट इज दैट यू मस्ट अंडरस्टैंड व्हाट यू नीड एंड गिव प्रायोरिटी टू इट नीड को भी जानते हैं लोग तो प्रायोरिटी फिर नहीं देते and उसके लिए we have to work. So my dear young people, you have opportunities. Luckily, you are not under that pressure as we were when seeds were being imported and we didn't have money to buy. लाल गेहूं खाया है हमारे प्रधानमंत्री ने कहा एक दिन हफ्ते में ना खाओ ऐसे भी दिन आए इंदिरा गांधी ने कहा कि अपने प्राइम मिनिस्टर से बाहर भेज करके भीख मत मंगवाओ. When she went to USA, she was refused with the support for wheat when we were needing it before just green revolution. That's how the point is that you have lot of again opportunity. So we must understand what we want. We need to take care of our nutritional security. Therefore, not only the disease, and now the climate is telling that we have to breed for the abiotic stresses. तो abiotic stresses के लिए traits क्या है, उनके लिए evaluation, उनका characterization. इसी तरह से nutrients के लिए क्या है, अब आजकल जैसे जिंक विंक का होने लगा है, तो इनके ऊपर प्रायोरिटी देके आप नेटवर्क में evaluation का प्रोग्राम हो, ऐसे ट्रेंड लोग जो हो रहे हैं, इनके पास फैसिलिटीज हो, उनको आप इन्वॉल्व करें. And then there had been the last point, because time is short, they may have questions and some discussion. कि जब से हमने ये सीखा है कि जेनेटिक रिसोर्सेज हमारी धरोहर हैं, तब से प्रॉब्लम शुरू हो गई. जब तक हमने सोचा था जेनेटिक रिसोर्सेज आर फ्री फॉर एक्सचेंज एंड दे आर कॉमन हेरिटेज ऑफ ह्यूमन काइंड, तो हम देते रहे लेते रहे, आदान प्रदान चलता रहा. जब से हमें बताया गया, तो बताया तो इसलिए गया था कि भाई जो आप, अगर आपकी बासमती राइस है तो आप उस पर जीआई कर लो, आप उनको अच्छी तरह से प्रोटेक्ट करके अंडर सर्टेन कंडीशन दो, ताकि कोई बेनिफिट ले रहा है तो शेयर करे. तो ये नहीं कहा था कि बेनिफिट ही ना दो, आप किसी को शेयर ही ना करो. तो वो एक माइंड सेट हमारा जो है वो ब्यूरोक्रेटिक हो गया, या वो औरों के हाथ में चला गया, पॉलिसी मेकर्स के हाथ में चला गया. पंचायतों को हमने कहा कि तुम्हारा है, आप पंचायतों से भी परमिशन लो. बायोडाइवर्सिटी अथॉरिटी का एक्ट बनने लगा, स्वामीनाथन साहब ने उसको डलवाया. मैंने कहा डॉक्टर साहब इसको मत डालिए. तो कहते नहीं, वी हैव टू स्ट्रेंथन देम. हालांकि उनको अभी तक रूल नहीं दिया गया है, पर उनको अवेयरनेस आ गई है कि ये मटेरियल हमारा है, तो हम चाहे लोगे तो हमारा भी इसमें हिस्सा होना चाहिए. तो ये प्रॉब्लम्स जो है मैग्नीफाई जब हो जाती हैं, और उसको अगर समझा ना जाए कि वो किस लिए किया गया था, तो उसके रिपरकशंस काउंटर प्रोडक्टिव हो जाते हैं. तो वो काउंटर प्रोडक्टिव ना हो, हमारा यही रोल रहे कि हम आगे अपने इंटरेस्ट में जो कुछ कंस्ट्रेंट्स हैं, because Pratibha must have given the lecture already about all these laws and biodiversity, diversity and all those, and कैसे हम कहाँ इधर से कहाँ पहुँच गए, एक्सचेंजेस में उल्टा घट गए तो बढ़ रहे थे. तो we have to now see how do we take care of all these, and in the process see that we utilize them. The last point which is there is about utilization. Unfortunately, it is not the NBPDR, it is overall globally that efforts in plant breeding to utilize more of germ plant has declined. We have become more dependent on international centers and pre-breeding material. तो आसान काम मिल गया ना भाई, वहां से प्री-ब्रीडिंग मटेरियल आ रहा है, एनबीपीआर इंपोर्ट करके दे रहा है, कोऑर्डिनेटेड प्रोग्राम्स हैं, तो इवेल्युएट करवा रहे हैं, उसी में से कोई अच्छा निकला उसी को मैं, अब मेरी तो गाड़ी बन गई, एक वैरायटी का नाम निकल गया, मेरा तो काम चल गया, आगे कुछ हो या ना हो, मैं प्रोफेसर भी बन गया. The question is that in the whole process, now the utilization, I remember when I was young scientist, genetic resources के ऊपर सबसे ज़्यादा जोर था, और variability नहीं है, और लाओ, और unless you have genetic variability, unless you work on it, and unless you try to improve it, you really cannot make that impact. So utilization part के लिए even globally एक प्रोग्राम चलाना पड़ा FAO को, which is known as Global Initiative on Plant Breeding, GIPB, supported by Bill and Melinda Gates Foundation. आज वो करीब 15-20 साल से चल रहा था, प्रोग्राम चल ही रहा है, और वो इसलिए कि भाई जेनेटिक रिसोर्सेज को और यूटिलाइज करें। और इस बाद में हमने ये जो कंसोर्शियम भी बना इसका, व्हाट यू कॉल इट, जो जीन बैंक को सपोर्ट देती है,
क्रॉप ट्रस्ट,
क्रॉप डाइवर्सिटी ट्रस्ट जो है,
GCDT.
GCDT, तो GCDT को भी I was involved, so I don't want to give on all those details. तो हमने ही उसको क्रिएट करवाया, 100 million का सपोर्ट एज ए चेयरमैन ऑफ GFAR. I have been chairing all those meetings with the donors, with the CG centers participating. कि भाई ये नहीं दोगे तो कैसे चलेगा। उसका था कि सपोर्ट जीन बैंक्स को नेशनल सिस्टम को जाए, और वो चली गई और डायरेक्शन में। सपोर्ट सारा जो चला गया इंटरनेशनल जीन बैंक्स के लिए, जबकि पर्पस था कि जिन नेशनल सिस्टम के पास जीन बैंक नहीं है। और इसीलिए जब मैं वहाँ सेंट्रल एशिया में गया, मुझे अपॉर्चुनिटी मिली वर्ल्ड बैंक के प्रोग्राम के तहत, उसमें CG सेंटर साथ में थे, तो there was no such objective, no such program, but out of my own resources सब लोगों के यहाँ हमने जीन बैंक एस्टेब्लिश कराई। तो क्या 2-2 साइंटिस्ट, 3-3 साइंटिस्ट पूरा नेशन, क्योंकि वो सब इंडिपेंडेंट हो गए थे रशिया से। When they became independent, because they had their gene bank in Vavilov Institute in St. Petersburg, they didn't have, जबकि जर्म प्लान सारा यहाँ इनके पास था। तो अब इन जर्म प्लान को कैसे संभालें, अब ये इंडिपेंडेंट हो गए, इनके पास जेनेटिक रिसोर्सेज को संभालने का कोई नहीं। तो नेशनल सिस्टम को कह के 2-2, 3-3 साइंटिस्ट असाइन कराए, उनके 2 कमरे खुलवाए, उनको भी मतलब जो है एयर कंडीशन किया, उनके रैक लगवाए, उनके साथ वो बॉटल्स में फीड रखते थे, उनको बॉटल्स के बजाय जो है वो पानी वाली बोतल को आधी काट के। That was the kind of gene bank system which we had in Uzbekistan, Kyrgyzstan, Tajikistan, Azerbaijan. So the point is that we now have to use more of these genetic resources, and the emphasis today is that even in the last, जो हमारा Delhi declaration था, कि emphasis should be on greater use of crop wild relatives. आपके कलेक्शंस में अगर देखा जाए तो crop wild relative only 1% है टोटल कलेक्शन।
113%। अब तक 13-14 हो गया।
13 हो गया
जी।
पता नहीं आपके डेटा में पहले का होगा, ठीक करवा दीजिए उसके डेटा। तो we have to therefore correct this also, and people should know what we have. और जो हमारे पास जल प्लान में इसको इवेल्युएट कराइए, तो regenerate कराइए, make sure that there are no duplicates there. Lot of challenges there. The work is not finished by इलाके अंदर रखने से। Now अंदर से हम क्या निकाल सकते हैं, that is another challenge. All these trainees who are there, they should get our partners, they should take responsibility, and on whatever crop they are working, they must try to see what can more be done. So with this, I would like to see, it is good that NBPGR has organized this course. I would like to congratulate Dr. Deepu Singh, also Dr. Gautam, and Jyoti Kumari and Dr. Huda for having taken pains to organize. But this should not be, end in itself. This should be beginning for strengthening genetic resource activity at national level. And with you as 80 participants, now we must say that we have added 80 more ambassadors of NBPGR. They must, and that linkage should continue. You must have a kind of, group formed and keep on exchanging information. In future, whatever your problems are cannot be all solved, but at least the exposure is there. And unless you try to know, and farmers even say that I don't need subsidy, I need good knowledge. Even young scientists would like to have good knowledge. It is not information की कोई कमी नहीं है. Information तो बहुत है, उसकी flooded है. और अब तो AI आ गया तो इसने तो और नाक में दम कर देना है लोगों का. गूगल को दम कर दिया. अभी तक तो गूगल को अच्छा मानते थे, अब तो गूगल भी परेशानी में आ गया. So this kind of information, पर information को knowledge में convert करके और उसका ज्ञान हमें मिलके उसका फायदा लेना is actually what we should aim at, and that is what is going to help us in our future career. So not think that genetic resource activities are not important, not scientific. They are the basis for the future of our young generation. They are the building blocks for agricultural growth, and genetic resources are likely to help a great deal in addressing the existing challenges of sustainable development goals, nutrition security, climate change, and also COVID pandemic. So that's what is our strength, and we must get much more activated and catalyzed after this course to continue contributing maybe more than what we were doing before, and possibly that would be the positive outcome of this particular training. Thank you very much.
Thank you very much, sir. In the meantime, if the arranges to have people asking a couple of questions, I must take this opportunity to thank you, sir, because when I joined the Agricultural Research Services, you were the Director General and Deputy Director General at that time, and we from that time on. So I am, and also my colleagues Kailash were in the same batch. We mesmerized with your personality, cult, and more we interact with you, we learn many things. I am probably the greatest of the learners here, because I not only have to run the administration, but also have a thorough knowledge of PGI. I must take this opportunity to assure you that at several occasions you have flagged certain issues, right from first Agro-Biodiversity Congress, which I participated as a director of the Indian Institute of Wheat and Barley Research. Since the last symposium we had in Delhi from ISPGR, several issues are now being addressed. You mentioned about permafrost and all those activities. Also, the revision of the guidelines for registration. 1 or 2 rounds of meetings have already taken place. And National Active Jump last night also, we have initiated action into this, and it will be visible very shortly. We have mentioned a lot of things. It is not only beneficial for the participants of this training program, but also for people like us in NPPGR for shaping the direction in the coming time, because perspectives are changing. What actually you sir initiated at that time, probably the perspective has changed a little bit, and therefore we need your guidance all the time. Jyoti, have you arranged anybody asking questions? Couple of them.
Sir, they last check.
So thank you very much, sir. I couldn't take much time in acknowledging the support you provide to MPPGR. Thank you.
So due to shortage of time, dear participant, we will take up only few questions. So I may call upon Dr. Tarak Nath Saha. You may ask.
Thank you, madam. Most respected Dr. R.S. Paroda, sir. Sir, we feel blessed listening to you always, sir. And it is an opportunity for us to listen to you also, and many things we also came to know. And also we are thanking to our director, NBPGR, for taking this nice training, which it is just many things we are learning, sir. And in this training, sir, actually, sir, I have a question, sir. Sir, actually in Directorate of Floriculture Research, we are having around 140 plus germplasm of chrysanthemum, besides roses, gladiolus, jasmine, and other crops. Most of the, I'm working on gladiolus, I mean say chrysanthemum, and most of the varieties are from NARS system, sir, and SAUs, and some are from private. And few of the material we are also having that we have collected from our local sources or from nurseries or from even other places. Recently, we have evaluated all our germplasm for essential oil and pigments content. And we found some of the lines are having very promising and possessing essential oil, which are having anti-mosquito and some other repellent property activities.
The questions
So
would be sorted.
my question is like this: how to get here how the IPR issues will be addressed in that, because we do not know the source of material and who has bred that material, sir. Sir.
He says lot of collections of flower, particularly resin,
Chrysanthemum.
and they have evaluated for oil content in that. So we are thinking about the IPR related issue to protect that. उसके लिए तो स्टैंडर्ड प्रोसीजर है मैडम, आपके लेक्चर में बताया होगा सर. We'll take this.
If you have some of those lead materials, firstly you can get those registered. So one part and get those evaluated as well, and make sure that they are available in the national collection wherever it is maintained. They are then maintained also. I was yesterday only, first time in my life, went to Mughal Gardens. I had been going there every year as Director General on 26th January to celebrate with the VIPs and the guest of honor for the country, President, Prime Ministers. But then that was over the tea in the afternoon. So never had an opportunity to see what Mughal Garden, now it is changed, the name is changed. So somehow my wife said, why not we go there today? I said, yes, we have not seen and weather is good. वहां जाके देखा एक वैरायटी पे लिखा हुआ था डॉक्टर पी पाल रोज. मैंने कहा और भी मिलेंगे, फिर रोज गार्डन भी था, बड़े रोजेस थे, कहीं नहीं मिले. IRI में डॉक्टर बीपी पाल ने इतने सारे रोजेस निकाले, मेरे ख्याल से मोर देन 20-30 होंगे. एक कॉर्नर भी होता था, मैंने अपना घर भी दे दिया. और मैं हर बार हर डायरेक्टर साहब से रिक्वेस्ट करता रहा कि भाई उन रोजेस को तो वापस मेंटेन करो. आज अगर आप चाहें तो सारे रोजेस आपको नहीं मिलेंगे. दिस इज ए फैक्ट. ओके, सो द क्वेश्चन इज a genetic resource person. You have to make sure that whether it is being cultivated or not, once if it is good for certain purpose, whether oil or any aroma or even गुलकंद भी बनती है रोजेस की, तो और पान मसाले में भी डलता है, तो इनकी अलग अलग क्वालिटीज हैं और उनके अलग अलग एरोमा है. कई बच्चे उनको फूलों को सूंघ रहे थे, कहते हाँ इसमें खुशबू है, इसमें नहीं है. इट वाज एन इंटरेस्टिंग एक्सपीरियंस यस्टरडे. I'll be there. आप नहीं गए हो तो आप भी जाइए एक दिन. It's worth going because genetic variability उन्होंने रखी हुई है. मेडिसिनल प्लांट का गार्डन है. डहेलिया मैंने ऐसे कभी देखे नहीं, तो मैंने कल वहां पे देखे. तो tulips are there. So the question is that enough can be done. और हम एक कल और परसों on floriculture पे एक नेशनल डायलॉग भी कर रहे हैं कि ये फ्लोरिकल्चर का जो यहाँ पे कोई इंटरेस्टेड साइंटिस्ट हो तो पार्टिसिपेट कराएं. तो आप जो कह रहे हैं वो ठीक है कि इनको अगर इवेल्युएट किया है तो ना कोई टिश्यू कल्चर जीन बैंक है तो वहाँ पे इनको सारे डोजेज होने चाहिए थे. आपके टिश्यू कल्चर में ले आओ भाई. अगर IRI नहीं कर रही तो सारे रोजेज की एक वही रिपोजिटरी कर दो. डॉक्टर बीपी पाल के नाम पे, क्योंकि he was the promoter of genetic resources. उन्होंने हरभजन सिंह जी को एनकरेज करके इंट्रोडक्शन डिवीजन बनाया था, और फिर जब डॉक्टर स्वामीनाथन डीजी बन गए थे तब उन्होंने इसको इंस्टीट्यूट बनाया था। तो जेनेटिक रिसोर्सेज के इन लोगों ने बहुत सपोर्ट की है। तो पाल साहब के जो मटेरियल है जेनेटिक रिसोर्सेज का, वो सारी वेराइटीज है, वो भी कहीं एक जगह तो करा लो। आई होप दे आर देयर इन IRI, आई एम नॉट अवेयर अबाउट इट, सो बट वन शुड टेक स्टॉक ऑफ इट। आपने जो पॉइंट कहा वो ठीक है, और हमें अब न्यू ट्रेड्स की तरफ जाना चाहिए। आई मेट ए फार्मर इन हरियाणा, इसे मैंने कहा भाई क्या करते हो आप, बड़े प्रोग्रेसिव फार्मर लगते हो। कहते जी मैं रोज उगाता हूँ, मैं कुछ नहीं, धान गेहूँ नहीं उगाता हूँ। मैंने कहा रोज उगाते हो तो रोज का क्या करते हो? कहते जी रोज का तेल निकालो। मैंने कहा तेल क्या करते हो? कहते जी मैं तेल इंडिया में नहीं बेचता, मैं UAE में बेचता हूँ, और बाजार मेरे पास आता है, मैं बाजार के पास भी जाता हूँ। तो उस टाइम में वो 2 लाख रुपए का 1 किलो तेल बेचा। सर्व अरब के जो रईस लोग हैं इनको लिंक बना हुआ है। द क्वेश्चन इज दैट एनी प्लांट यू टेक एंड द रोज कलेक्शंस बेस्ट आई हैव सीन इन रांची, that is the Women Rose Association of Ranchi. The maximum variability, Dr. Anil Singh वाइस चांसलर थे उसमें, और वो वहाँ पे उन्होंने रोज शो किया हुआ था, तो आई वेंट देयर। तो ली रही मिसेस नायडू होती थी उनकी प्रेसिडेंट। ऐसे रोजेज मैंने दुनिया में नहीं देखे। तो हमारे रांची में इन्वायरमेंट होगा हम आके, बट द काइंड ऑफ वेरिएबिलिटी, और ये आप डॉक्टर पाल साहब के घर पे ही वो एक बेल रोज की है जिसमें छोटे छोटे व्हाइट रोजेज लगते हैं, वो बेल ही कहीं नहीं मिलती हमें। Scope is too much, but then effort is required and consistent effort.
So,
Thank you,
sorry,
sir.
dear participants, although sir was very much interested for taking questions, but we don't have sufficient time. So please put your questions in the chat box. So that will be answered later by the sir. Really, sir. Okay, okay, sir. With your permission, Dr. Reena Mehra, please have the quick question.
Good afternoon, all. Thank you so much for giving an opportunity to interact with Dr. Baroda, sir. Sir, I have a very small question that I am from ICADA, and we have also kept a black box in NBPJA, and almost 12,000 accession of lentil is there. Sir, is it possible to characterize those material at our center? If yes, then what would be the procedure to get this? Thank you.
I think you will.
Yes, sir, she said that we have kept 12,000 accessions in the black box kept in NPPGR. You want to characterize?
उसमें क्या what is the difficulty?
No problem.
Collaborate करके plan कर लीजिए, locations identify कीजिए, and no doubt you will require funding support. So if there is funding support, okay, otherwise prepare a project, get it approved, and only thing is that there should be something uniqueness in terms of evaluation part.
Yes.
Not clean evaluation नहीं होना चाहिए. So specific trait उनकी क्या value है उसके ऊपर. Actually, you said you are from ICADA?
Yes. Yes.
ICADA. तो ICADA तो आप थायरस का कलेक्शन को इवेलुएट करा लो ना सारा. इतना इम्पोर्टेंट है. और जो आपकी वहाँ की वैरायटी थी जो थायरस फ्री थी, तो उस वैरायटी को इंडिया में लोगों को और देकर के ब्रीडिंग प्रोग्राम में उसको इंटीग्रेट करा लो.
Yes, sir.
You have such a good germplasm also of this lentil.
Yes.
Lentil is a very important crop for rice fellows in the eastern region.
Yes, sir.
Germplasm all लाइए और दीजिए. Only thing is that again, not only bold or small seeded, but the trait relating to the protein quality, and productivity, etc. So ICADA has sort of Papa Bean लिया है
Yes, sir.
Papa Bean so important. हम खाते थे जब बचपन में छोटे थे
जी
और राजस्थान में मिलता ही नहीं है बाकला कहते थे हम बाकला है ही नहीं
Bakla, yes.
It's a part of ICR Sikada MOU, and we are taking up Fava Bean Lentry.
तो आप कोलैबोरेट आईपीपीआर डायरेक्टर साहब से बात करके आप शिव कुमार और आप लोग आपका भी सेंटर है तो चाहे और सेंटर या इनके के सेंटर इवैल्यूएशन हो सकता है पैसा आप लोगों के पास लकीली थोड़ा है इंडिया ने दिया है या इंडिया के पैसे से ही चलते हो आप लोग
का मटेरियल भी लगा है सर हमारे यहाँ पे इनकी एक नर्सरी भी आई है सर काफी
आइडेंटिफाई कीजिए प्रायोरिटी और इम्पोर्टेंस ऑन बोथ साइड्स एंड पार्टनर्स को इन्वॉल्व कीजिए
यस
क्रेडिट दीजिए सबको
सर ये जो ब्लैक बॉक्स अरेंजमेंट की बात कर रहे हैं आई थिंक सम पीपल डोंट अंडरस्टैंड ब्लैक बॉक्स मतलब बंद करके नहीं रख दिया है तो दोनों इनका रखा हुआ है उन्हीं की परमिशन से बाहर निकाल सकते हैं इट इज ओनली ICADA कैन टेक इट आउट और फिर यहाँ पे
पास जो है
हाँ सर एनपीपीआर में जो ब्लैक बॉक्स अरेंजमेंट है जिसका रखा होता है उसकी परमिशन चाहिए निकालने के लिए तो ये
वो
नहीं से आना चाहिए
तो फिर आपस में दोनों इंस्टीट्यूशंस की बात है
यहाँ पे बात करने से बेहतर विल डिस्कस दैट शिव कुमार ओके परमिट वन मोर क्वेश्चन सर बट देन
ओके कीर्ति रानी
और सीजी सेंटर्स का मटेरियल भी ब्लैक बॉक्स बन रहा है
एक सेफ्टी डुप्लीकेट
सेफ्टी डुप्लीकेट तो ठीक है अगर वो चाहिए
तो उसमें तो कोई प्रॉब्लम नहीं ओके कीर्ति यू मे आस्क क्विक क्वेश्चन
A very good afternoon, sir. So I have one general question. What, as a young scientist, what I feel that we are more focused or inclined towards more number of publications, but there is always a gap in what we publish, what is on piece of paper, and what is, they have practical implications. So, sir, what would be your suggestion for roadmap or a way forward, way ahead? For a young scientist like us, so that we can play a role for a secure and sustainable agriculture growth.
Well, I think I would not say that publications are not important, and if you are publishing or any scientist is publishing, it is good, but it's his capability to express whatever is being done more scientifically for benefit of others. But our focus should be also for the society, as I told in the beginning. And if it is for society, then what are you giving back to them? One is for your own, promotional avenue is important, but back to society. And in that respect, if you Dr. Warlock, you would have asked, because once I had discussion with him, that you are seeing change in CGIR. Since your time, the current time in CGIR is much changed. What is your feeling about it? So his just blunt answer was, if I were scientist today in CGIR, I would have not given any single variety, but I would have published papers. This was his answer. I think it gives all the message. You don't require to be elaborated. And he says, wherever I go in the institute, I see everybody sitting on the desk and just, what are you doing? Writing either project proposal or writing report. तो जब donor driven program होंगे तो उनकी रिपोर्ट ही बनेगी ना, या साइंटिफिक पेपर होंगे कि मैंने इतने पेपर निकाल दिए. ये तो कोई नहीं कहेगा कि तुमने वैरायटी दी नहीं दी, और उनके ऊपर कोई प्रेशर नहीं है कि वो वैरायटी दे या ना दे. So that therefore you need maybe people like Warlock, but also people who can publish and benefit the scientific society and themselves.
Thank you very much, sir, for sharing your vast knowledge and experience about PCR management and utilization. So your encouraging words always help in boosting our intellectual strength. You have been the all-time mentor of the MBPGR, sir. So we are very grateful to you, sir. So now we would like to felicitate our sir. ये हमारा सौहार्द है, it is our great. इससे तो लगता है कि मैं यहाँ का पार्ट नहीं हूँ. When you came and you said कि lecture देना है, मैंने कहा तो order है भाई. This is not request. Very much, sir, for accepting our request, and thank you, our director, for making this happen, sir. This event. Thank you for your affection. For spending your helpful time, sir. Thank you very much, sir.
2 minutes में बोलती हूँ.
Now I would like to Dr. K.S. Huda to propose formal vote of thanks. Please, sir, Dr. Huda.
Good morning. And I am very thankful to today's chief guest, for the motion, Dr. Raj Prahodaji, who has been with us with a short notice, and he will after changing the dates. And sir, briefly, in one line, I would say that you have educated us regarding the PGR, growth of NPPGR, and how to evaluate and utilization of PGR. I'm very thankful, sir, for sparing time from your busy schedule. I'm also thankful to all HODs, OICs, unit in charges, and my team members who are actively involved since February 1 in this training. And we will fulfill your expectations through this training. Thank you very much once and all to all. Thank you.
ठीक है. आज एक प्रोडक्ट. और हम लोग पीछे खड़े हो जाएंगे. ये किधर है? अरे मुझे नहीं सर, खड़े हो जाते हैं सर. हाँ ठीक बात, खड़े हो जाते हैं. हमारे चलते हैं. हाँ बस यहाँ भी ले रहे हैं.
यहाँ तो यही हो ना, बाहर ठीक है. तो अरुण लंच के लिए
resistant accession in various pulses and vegetable crops. He has published more than 23 research papers in reputed journals and authored 8 books and written 8 book chapters and more than 20 technical and popular articles. He has several award and recognition and got best poster awards, best oral presentation awards in international conferences, and he received Professor DJ Rakshi Academic Scholar Award from Nematological Society of India and Young Scientist Award from SBER Society, Tripura. He is also involved in teaching of various courses related to plant quarantine and nematode management of the postgraduate school of IRI, and he also guided many students. Thank you very much, ma'am and sir, for joining with us for in this training program and for delivering the lecture. And now to use, ma'am, please share your slides and video and share your knowledge with our participants. Ma'am, please.
Thank you very much, Subhash, for the very nice introduction. Dear participants, I'm going to talk about mainly entomological aspects which we are dealing with in the plant quarantine. So plant quarantine is a multidisciplinary subject, and we have we have entomology, nematology, virology, plant pathology, weed sciences. All the experts are available in the plant quarantine division, and all of us work together towards one common goal, which is to make the material which we are importing free from pests. Our target is only that. So let me start sharing my screen. Let me tell you initially about briefly about the various techniques and what are the commonly intercepted insects which we are dealing with. And thereafter, I will be showing you a couple of videos so that you can have an idea as to what and how we are doing the work. So let me start sharing my screen.
Presentation mode.
Let me go and make it presentation mode. Is it full screen now?
Yes, visible.
Yeah. So this is just the practical part of it. I'm sure the theory must have already been told by Dr. Celia about the legislations, what is PQ order, and how we go about doing the plant quarantine. And briefly, she must have also told about joint inspection and thereafter specialized detection, how we are doing for various kinds of pests. So for entomology, we are basically, when we import, we have two kinds of materials. One is perishables, which is vegetative material, and durables, which is cereals and storage agricultural commodities. So our methods for detection and the kind of pests that we detect in these two commodities will be quite different. That is why I have kept this slide. So when you talk about insects and mites, based on their habits, insects are one of the largest groups of known fauna, which has diverse habits. A lot of different types of mouthparts, 6 different types of mouthparts are there. But here, when we are talking in quarantine, we are dealing mainly with the sucking type, where we are kind of intercepting pests in vegetative material, like aphids, bugs, scales, whiteflies, or thrips. And the chewing type, which are associated with both seed as well as vegetative material, that is beetles, borers, caterpillars, grubs, maggots, worms, all these kinds of pests. Another category is, this belongs to a different class altogether, that is mites. Mites belong to arachnida. They are small, very tiny. Sometimes you can see them with naked eye, otherwise mostly they are microscopic. So they can be, adults can be there, or even immature stages, eggs, galls, which they cause, and all these kinds of pests are there associated with the. So when just a little bit, because I have come to know through Jyoti that the group is quite heterogeneous. So just to give a clarity on the insect structure, insects have a segmented body. The body is divided into 3 parts: head, thorax, and abdomen. As a generic structure, I'm saying, there may be variations. Then compulsorily, they would be having 3 pairs of legs. In some, and there may be modifications. They are tracheated. Trachea is there for respiration. Usually have a pair of antennae and 2 pairs of wings. They may be modified or they may be present as 2 pairs. So this is the insect structure. And when we look at mites, they belong to class Arachnida. They have 4 pairs of legs. Generally, again, there may be variations. Then body is divided into generally 2 segments, cephalothorax, where head and thorax are fused together, and an abdomen. And this is the basic difference between insects and mites. When we look for the general symptoms of insect damage, either the whole seed could be consumed and reduced to dust, or internal contents of the grain are eaten, leaving only the outer shell, only the seed coat is remaining, and the insect is feeding entirely on inside. Or the germinal portion is scraped and devoured, and
This would result in the seeds not germinating. Then scouring of the surface of the grain or burrowing into the holes, tunneling, webbing together the grains. So various types of general symptoms you can see when, and this inspection is done, or detection for insects, presence of insects is done both at the country of origin while issuing a phytosanitary certificate by the exporting country, and at the destination here in quarantine. When we receive any foreign material, we all examine and look for presence of insects and mites. So infestation may occur when the seeds get infested while developing on the plant, or they get infested at the threshing ground, during transport, or during storage. And they are broadly categorized as external feeders or internal feeders. So external will be feeding externally, multiply even in stores and cause serious losses. Whereas internal feeders, they will not show any external sign or symptom of their presence, but they will be continuously feeding inside the grain. And once they emerge, then only you realize that the seed was infested. So they are very, very dangerous. So when we see the visual examination, in visual examination, it will give you at least 60% quarantine security. The moment you open a packet, the very appearance of the seed, the very smell of the seed, the very look of the seed, you can make out that this doesn't look healthy. So the inspection efficiency depends on the skill and experience of the quarantine person. So it is very important. Visual examination gives you 60% of quarantine security. You can detect external feeders. You know where to look for, in the cracks, in the crevices, in the envelopes, in the corners where the seed, the insect might be having. And then you use a naked eye, or you aided eye with a magnifying glass or illuminated magnifier or a binocular. You can go and look at the various insects. So we have one technique which we are using for detection of hidden infestation, which is X-ray radiography. This is a very, very efficient system. This machine that I'm showing you, I will be showing a demonstration of this also. This is a real-time X-ray machine, and basically the X-rays that we are using are not damaging. The range at which we are using, they are not damaging to the germination of the seed. That is why we are using on germplasm. It is a non-destructive technique. It is, once you purchase this machine, it's a one-time investment. And then after that, you keep using it. It's a real-time X-ray. You don't need to develop, no consumables are required. Only you need to keep using all the precautions. Sufficiently rapid, then it is quite reliable. You can magnify, look for various stages. And this technique indicates not only the insect inside, but also many other seed parameters also, seed filling and all. We are routinely helping our colleagues in the conservation division, evaluation division, for doing other applications using X-ray. So when we are looking at the cycle of development of bruchids, the egg stage is on top. You can see it with the naked eye. The first instar larva will bore inside the seed. And thereafter, second instar, third instar, fourth instar, and fifth instar, and pre-pupal stage. Pupal stage is all inside the seeds. And this first instar larva, once it enters the seed, will plug the hole. So externally, you will not see any sign or symptom of the presence of the insect inside. And only when the adult emerges, you can see this in the picture. This is the operculum stage. When the adult emerges out, then you see a big hole. Then you realize that the commodity is infested. And now I will be showing you X-ray radiography video. Let me stop sharing this and share my video. Share screen. This one. Can you see my screen?
Yes, madam.
Yeah. So this is the X-ray machine. He is our technical officer who is going to do the X-ray in front of you. And this he's telling about a little bit about the machines. This is an old machine where we are using X-ray film. Here we load the film in black envelope and then keep the sample on top of the loaded envelope, expose it to X-ray, and thereafter we develop the film. And then you can see. But this Cooptex expert machine, this is a real-time X-ray machine where you can just keep the sample inside, and immediately the image will come onto the screen. And we have a list of 344 plant genera. This list is showing. This is the list with which we tally. So this is 344 plant genera of the plant material which are known to carry hidden infestation. So this is a literature-based information that we have compiled over the years and updated it from time to time. And whenever we get any material from this list, we will X-ray it. So this is the record X-ray sheet that he is showing, where you will enter all the information from where it has come, what is the crop, what is the IQ number, how many samples are there, and thereafter you switch on the X-ray machine. It takes a while to start because it has to warm up and then calibrate, and only then after once it is calibrated, we will. So for calibration, this is the lead shield that he is placing on the detector. So this exposure area is covered completely with this lead shield, and then he will switch on the calibration mode. And thereafter, once it is calibrated, he will remove the shield and keep it back. And now the machine is now ready to use. So he will open the software. You can see the screen. You need to hear again. It is saying that the machine is calibrated. So all the seed material, whatever is there, he is now showing. See how he is going to plate. The seeds have to be arranged in a single file, in a single row. So none of the seeds should overlap, and they are set in such a manner that they will follow a particular seed geometry. So he is making the X-ray plate. And now he's going to label it. He's putting one alphabet. See, A. So this is A sheet. So that once you expose, you would not know which is which. So in a sheet, you will note down all the information. A is your sample number, so and so. B is your sample. And there is a inbuilt log inside the machine. So here he will fill in all the data. So this is a domestic quarantine material. So DQ 189 2022. And this is cowpea. So he will fill in the data. And then in the notes, he will put how many samples? 10 samples. And he will write from where it has come. So he will put in the notes where it is from MP. It is from MP. So he is writing, and this is saved. So once he has filled in the information for one DQ, now he can keep taking images, and the images will be stored in the same folder. So the plate is kept right in the center. And he has shut the X-ray machine, and now he is going to start the X-ray. See? So to acquire the image, so he is going to click acquire, and immediately the red light will come in the machine. That X-ray power accelerating. So now it is X-rays are showing. So you can see in the machine, all the 3 lights are on now. X-ray is on. So images will now be shown on the screen. Yeah, here. Now you see the image. You can see almost 80–90% of the seeds are all infested inside. And you had seen earlier, no? Some of the seeds are deformed. So they are showing some sort of curvy type of thing, but they are not infested. So now he will take out, without disturbing the seed geometry, he's going to take out the tray from the X-ray machine. And with a forceps, he will keep looking at the screen and tallying his sample. And he will left to right or right to left, he will, based on the geometry, he will pick the infested seeds. This is a very delicate and time consuming, as well as exercise which requires a lot of patience. So 4th seed is found to be infested, so he has picked out those. This is a heavily infested seed material, but when you look at the seed, it doesn't appear to be that bad. So after we take it out, we usually soak it in water for 24 hours. And thereafter, we will remove the, so he has already soaked some seeds, he is showing you. And here with the needle and forceps, he's going to tease the cotyledons and remove the insect. So he is going to remove the insect. And then we will take it to the other lab where we will mount it properly, dry it, and then examine under the microscope to identify it. And that part I will be just telling you in detail. So just see, he has taken out one insect. So he will gradually, without damaging the insect, he is trying to take out the insect. Because we need all the characters. We need the antenna, we need the legs, we need all the characters intact so that we can identify. See, you can see the insect complete. This is the Calissobruchus maculatus. But anyway, we will confirm with the key characters and look in the microscope after mounting. So this, I hope you could see the video. And now I will go back to my presentation. Yeah. So next is the seed transparency. Another technique that we use is X-ray. You can see that in X-ray, you are mechanically picking out the seeds. But many a times in the list, you can see in the 344 plant genera, we have certain seeds which are very small in size. And if you spread them in the plate and you try to pick from the seed geometry, you cannot do it. For such seeds, we use another technique, which is called as the seed transparency technique. And here, either in a strong alkali, you partially digest the seed, which become transparent, or we use lactophenol. Here, it is a mixture of 2 parts of distilled water, phenol crystals 2 parts, lactic acid 2 parts, and glycerine 1 part by weight. And this is a hazardous chemical, so we need to be very careful when we prepare. Always use a lab coat, gloves, so that it doesn't harm. And a person who is quite adept at doing all these things should be handling this material. So we will do the seed transparency, and once the seed transparency is completed, it is boiled in a water bath, and then it is taken out and observed under a binoculars. So this technique is a destructive technique. If we are getting, say, 50 seeds of a particular seed like trifolium, or 100 or 200 seeds, we will take a small pinch, maybe 20 seeds at random, and then do the lactophenol testing. If we find it to be infested, then we go in for fumigation so that we can salvage it. So this is a destructive technique, whereas X-ray radiography is a non-destructive technique where you do not destroy the sample. That's why we prefer X-ray radiography. And it is a dual purpose technique. It is not only used for detection, you detect as well as salvage when you pick the infested seeds, means you are salvaging. So it is a very foolproof and good technique. So let me show you lactophenol also. And stop sharing. Share my other video. Once here. Even. Can you see my video?
Yes, madam.
Yes, ma'am.
Okay. So now he is doing lactophenol testing. So he in the box he has trifolium seeds, which we got from some African country, and he has this lactophenol in the bottle for demonstration purposes only. We have already set the experiment. You can see 4 test tubes in the water bath, but now he is going to show you how he's made this lactophenol, and we keep it in a bottle in the closet, and then in the boiling tube, in a clean boiling tube, you take a few seeds from a small sample of the seed, maybe 20-30 seeds. Add it in the boiling tube. Now he will add the lactophenol, around 20 ml of that, and so that the seeds are completely covered with lactophenol. Yes. And then he will keep it in a water bath. After plugging it properly, he's going to keep it in the water bath. And in the water bath, he's going to let it remain for around 2 to 3 hours. And he will see in between to check whether the seeds have become transparent or not. So in between, when he will see, he will know that the seeds have become transparent, and then he will let it cool down a little. Now it's almost 3.5 hours. So the samples which we had kept in the morning have now become transparent. So he's just checking whether they have become transparent or not. So so some of them are still not okay. So he's checking the other sample. And in the glass Petri plate, you need to pour it in and then observe under the microscope. So he is taking the glass Petri plate. Now and he's going to pour this material into it. So one needs to be very, very cautious. If it's too hot, then you hold it with something which will, and shake it properly and then pour. Otherwise, the seeds tend to settle down. So let it cool down a little and then observe under the binoculars. So now he has poured it all. He's tapping the bottom to take out the remaining seeds. And this he will keep in the rack. And now he's going to see in the microscope. So you can see from the previous work also, he has kept few plates that he has been doing. So in continuation, we recorded this video so that we could show you. So he's going to observe the seeds. Some of them you can see some dark thing inside, no, and the transparent. So those seeds which look dark and having an insect inside, you can actually see the insect inside. I'll show you more clearly in the photograph. So this is, and thereafter, once you take it out, you mount it properly, and then I try to identify. And the whole sample, what you do is, once you have seen the insect inside, that the sample is infested, the remaining sample you fumigate so that the sample, if there is a live infestation, it will become dead, and you can give the healthy seeds or insect-free or live insect-free material to the indenters. So this is how you do the transparency testing. Here you can see almost all the seeds are okay. Only 2 are dark and having an insect inside. So anyway, our sample is infested, so we will need to do the fumigation. This again looks almost okay. Only I think there you can see 1 or 2 seeds which are dark and having the insect inside. So he is observing all the plates so that we know which are the insects, which are the insect infested material. So now I will stop sharing this and move on to the presentation. Can you see my screen?
Yes.
So in this, you can clearly see, these are the seeds which are observed under the microscope. And here, even the insect body parts you can make out. See the head, thorax, abdomen, the various parts, even antenna is visible. So the seeds become absolutely transparent, and then you can even observe the insect inside. So this is transparency testing. And now coming to identification, there are many types of identification. We can do the classical method where we use identification keys based on insect morphology. We have pictorial keys. We have digitized pictorial keys. And nowadays, even molecular taxonomy. We are exploring this area more because many a times we get insect stages, the larval or pupal stages inside the seeds, and we are unable to make out what insect it is. If we get an adult, we try to identify based on the morphology, but anyway, if we are getting immature stages, then it becomes difficult. So molecular taxonomy is still in a nascent stage here because there are many limitations for us. In quarantine, we are intercepting only 1 or 2 or maybe 10 insects. That too we do not know. And if we try to take out the DNA, so the quantity of DNA is too little, and many a times we are not able to use it for identification. So there are many limitations. Therefore, we are mainly depending on pictorial keys, digitized keys, and the classical method for identification. Sometimes if the we usually keep the immature stages in a bottle, or the seeds which are infested with immature stages in a bottle, try to see them through the developmental stages. Once the adult emerges, then we try to identify, to know what is the insect. So this is the, once the infested seeds are gathered from X-ray radiography, we soak them overnight. Next day, we will tease out the insect, retrieve it, and mount it on a card, label it properly, and then based on identification keys and reference collections, we also have a reference collection of exotic insects, all those insects which we have intercepted over the years. From 1976, we have a collection, and many a times we have to refer to them, to that collection to identify. And this is just the, since none of you are, any of you are entomologists in this, so you need to have a proper labeling, proper pinning. They would be knowing how to place the insect on the pin. We have special entomological pins which are nickel plated, which do not rust. And then how to label. You need to label from where you got, when you got, and who identified. Who is the collector? And then the identification on a separate label. So this is the type of detailed working that you need to have in mind. And this is the digitized keys that we are talking about, which we developed. This is an in-house work. Here we did, we prepared digitized keys. This is a database on all the available literature of bruchids in the world, more than 1,600 species covered in 71 genera. So this is, and for each, as many as we could, you can, it's a free resource which is available on NBPGR website, digital library on bruchids, you can access, and anybody can just simply register and start using. So here you, based on different characters, you put in 2-3 characters, and then on that basis, it's a unique combination of characters which will give you a identification. So you have identification up to genus level as well as identification up to species level. So these are few examples of various commonly occurring storage pests. This is Rhizophilus surinamensis, very typically having 6 tooth on the thorax on both the sides, projections. This is very, and the antenna resembles a club. So this is a very typical insect. Here, khapra beetle, most of you must be familiar, especially with the larval stages. It is a very, very important insect from phytosanitary perspective, because many of the countries do not have khapra beetle, and Indian consignments are routinely being scrutinized for presence of this khapra beetle. So especially going to Australia, to Vietnam, to Thailand, and many other countries. Then Plodia interpunctella, this is again a very commonly occurring storage Indian meal moth, which is occurring in the grains. Then drugstore beetles, Stegobium. Here there is another insect, Lasioderma. Lasioderma and Stegobium look quite similar, but the difference you can see, this has a 3-clubbed antenna, whereas Lasioderma here has a setaceous antenna. Plus here the body is quite hairy. On a broader level, I'm saying, when you see, but otherwise they are belonging to different genus and species, and they are completely different based on their genitalia, based on their other morphological characters. But for a lay person or a person who is not familiar with all these things, they can identify based on this. Then Cytophilus, we have 3 species of Cytophilus which are very, very important. Cytophilus oryzae, Cytophilus ziames, and Cytophilus grenadius. Cytophilus grenadius is not yet reported from India. Many of us mistake or misreport it in our reports. See, Cytophilus granarius is a quarantine pest. And it is uniformly colored and it has elongated pits on the pronotum. You can see. When you look at the comparison between the Cytophilus granarius, Cytophilus oryzae, and Cytophilus zeamase. Zeamase is slightly larger with very clear-cut markings on the elytra, whereas granary weevil, that is Cytophilus granarius, is uniformly colored. So these are small, subtle differences which distinguish different species. This is Rhizopertha dominica, again a very commonly occurring, most characteristic feature here is the head is deflects down. See, the head is just down, and this is the whole thing is the thorax. The thorax is completely covering the head. And this is Cytotroga serialella, which we are routinely intercepting. Then flower beetle is again in broken gains, or this is a secondary feeder, not a primary feeder. And Chrysobrookus, the broochids which we are intercepting are many, and many a times we are intercepting exotic broochids also from various countries. And this is Cystole coriandri, a chalcid, which occurs in coriander. We are routinely intercepting this also. Mango stone weevils, Sternochetus, another very, very important insect pests for which many countries have phytosanitary regulations. And this is a pest for which even US was not importing Indian mangoes. But once we had the phytosanitary treatment of 400 gray standardized for it, now they have also started importing. So this is again a very important pest from quarantine perspective. And now we come to the disinfestation. Once we have the infestation, what do? We remove it mechanically, we destroy, or we use various techniques or treatments. And this is a very important quarantine strategy, because many a times whenever we are getting material which is infested, our aim is to give the indenter some material, because nowadays exchange of material has become very important and difficult also. So whenever we are getting something, then we try to salvage as much as we can. Only when the material cannot be salvaged in any way that we reject. So these are the various regulations at the international level which deal with the phytosanitary treatments, mechanical, chemical, irradiation, feed, hot, cold, controlled atmospheres, and all. And these are the national regulations, insecticides rule, gas cylinders rule, prevention of food and adulteration rules and all that. And Bureau of Indian Standards also gives codes of safety for fumigation. And for any phytosanitary treatment, the major considerations are pest tolerance, whether the pest is able to tolerate it or will be killed by it, commodity tolerance, residue tolerance, and the duration for which the treatment has to be given. And depending on type of commodity, we do various types of treatments like mechanical, X-ray, radiography, fumigation, combination treatments, pesticidal dips and sprays, and aerosols. And when I talk about fumigation, it is a substance which is used in an enclosed space to kill an insect pest. It may be a gas, it may be a liquid, or it may be a solid. So gas could be methyl bromide, liquid could be ethylene, EDCT, ethylene dichloride, carbon tetrachloride mixture, or it could be aluminum phosphide or a crystalline powder like naphthalene. So you need to have special dose dispensers. You need, if it is a solid then or liquid, you need to have something that will heat it so that it will become gaseous. And here concentration time, exposure time, and temperature and pressure. These are the 4 things which are most important. So CTP is a constant. If you are reducing the concentration, then the duration of exposure has to be correspondingly increased. Then sorption, uptake of gas by the solids, penetrability, then temperature, permeability, and what are the equipments that you would be using. So these are important in fumigation perspective. You can have atmospheric pressure, atmospheric fumigation, wherein you fumigate the material at the normal air pressure. Or you can have a vacuum fumigation, where you need to have a specially designed chamber with reinforced stage, which is able to withstand the pressure. You remove the air pressure from inside, so the outside pressure will become too much. So you need to have proper pressure gauges and all. And this is the vacuum fumigation system that we have with us. And I will be just showing you a demonstration of that. Let me stop sharing my presentation. And just give me 1 second, huh?
Okay, madam. Okay.
Can you see my screen?
Yes, ma'am.
Okay. So this is
Yes,
two
ma'am.
of our colleagues who are sitting in the fumigation lab, and they are puncturing. See, these are all present. The sample is in the plastic packets. So they are puncturing the seeds, puncturing the packets so that they can, this is the vacuum fumigation chamber. The capacity is quite large. Here we can do a large number of samples. We have 3 fumigation chambers of different sizes. So this is the sample they are keeping inside now. They have punctured the plastic packets and have kept inside. And this will develop a pressure of, the vacuum is developed for one atmosphere. So now they are going to put the fumigant. Here they are going to use today EDCT, ethylene dichloride carbon tetrachloride mixture, which they would be using. So they are measuring out the chemical in the glass cylinder, measuring cylinder, and now they will be pouring it into the beaker with the cotton. And this beaker, this is a highly volatile and volatile liquid. So once it is kept inside in the enclosed space, it will evaporate and penetrate into the packets and kill the insect. So this they have taken almost 250 ml. So this is the amount that we would be keeping inside. Ethylene dichloride and carbon tetrachloride. So that is a mixture of 3:1. So they are going to see, they are keeping it inside, and now they are going to shut it and fasten it tightly. And exposure time or the treatment time reduces if you are keeping in a vacuum by what you are going to normally take around 24 hours or 48 hours. When you're doing vacuum fumigation, you can complete it within 5 or 6 hours. So that is the difference. So this is, so they are going to, they have C250 ml for 48 hours. So they are afterwards, this they are the next day, once they have completed the fumigation, they have opened, and now they will air the sample. They are going to keep it in the fumigation chamber, fumigation lab, on the table for a few hours, say 5-6 hours. and then they will be sent to the laboratory for dispatch. So this is the fumigation part which I wanted to show. So this completes my entomology practical. I hope you were able to follow.
Thank you, ma'am.
And
now I may request.
I hope I have not exceeded my time.
No, ma'am. You are on before time even.
Okay. So if you have any questions, I'd be happy to answer.
Ma'am, should we take a question
at the end?
At the end, ma'am. Is it
no problem.
Okay. for you?
Once Bharat also completes his nematology practical,
yes.
then we can have.
Then, okay.
Thank you. Thank you.
Now, I may request Dr. Bharat for share your presentation, ma'am. Dr. Bharat.
Yes, I'm sharing it.
Okay.
So it is visible?
Yes, it's visible.
It's full screen?
Yes, it's full screen.
Okay, thank you very much, organizers and Dr. Subhash, for giving us the opportunity for this conducting this practical in this virtual training program on the management and utilization of PGR. So I will be covering the plant quarantine processing nematology aspects, and we will be seeing the techniques which are used for the detection and identification of the plant parasitic nematodes from the samples which are meant for the exchange, whether it is import or export. So I will start my practical class with some introduction, as many of the trainees are not aware about these plant parasitic nematodes. So first I will these plant parasitic nematodes are significant constraint to the plant crop production, and which are causing around $173 billion losses worldwide. And if you see these plant nematodes, these are very small and multicellular organisms, and is found in all kind of the environments. But very little species are studied of these plant parasitic nematodes. Only 4,200 known species are there, and still many species are about to discover or not yet reported. So, but on the worldwide basis, if you see these 10 most important genera which are damaging to the agriculture and horticultural crops are this root knot nematode, which is coming under this Malodgyne genus, then Heterodera, that is cyst nematode, Globodera, a potato cyst nematode, and then there are many kind of the stem and bulb nematode, Diatolyncus. Likewise, Rhadophilus, Rotalinctus, Helicotalyncus are the most commonly known nematodes which are found to be associated with the crops of the agriculture and horticultural. And these plant parasitic nematodes are basically categorized into the two based on that feeding habit, root parasites and the aerial or the foliage parasites. So first of all, the root parasites. The root parasites, which are majority of the plant parasitic nematodes, are the root parasites, and they basically feed on the underground plants, that is roots, rhizomes, likewise with the parts which are present inside the soil. So based on their feeding sites, there are further categorized into the endoparasites, semi-endoparasites, and the ectoparasites. The endoparasitic nematodes enter inside the roots completely, and they feed and mature within the roots. Like most damaging nematodes such as Malodgyne, root knot nematode, cyst nematode, these are belonging to the endoparasitic category. In semi-endoparasites, they are partially penetrated inside the root, where anterior body part is or head is embedded inside the roots, while their posterior parts is outside the roots. While ectoparasites, they feed without entering the roots. They are freely move inside the soil, and they feed from the outside on the root exterior part. So another category, that is aerial or the foliage parasites. Most of the nematodes which are seed-borne in nature, they generally belonging to this category, that is seed gall nematode, stem and bulb nematode, then some nematodes which are belonging to the Angina family, the gall forming nematodes. These are basically move along with the seed material, true seed-borne nematodes we can call them, and some are the foliage parasites. So sometimes we get the other vegetatively propagated material also. So in that case, along with the leaves or the stem or the bulbs, other foliage parasites also comes. So if you see the examples which are examples of the nematodes which are introduced into the India along with the planting material are the potato cyst nematode, then stem and bulb nematode, and pine beetle nematode, burrowing nematode. These are actually detected. So sometimes detected. It is only one nematode is presented in India and it is under the domestic quarantine. That is potato cyst nematode, Globodera rustocanensis and Globodera pallida, which is introduced into the our country just after the independence, and then burrowing nematode which came to India through the banana propagating material from Sri Lanka. So these are the some examples which are spread along with the seed material. So during the quarantine processing, we have to very critical about the what are the pathways along with which the nematodes can spread. So there are 4 important pathways for introduction of the nematodes. One is true seeds, that is truly seed-borne nematodes are there. Then sometimes along with the seeds, we get soil or other media, and nematodes which are present in the soil, they can come along with this pathway. Then packaging material, some nematodes are infecting the wooden logs, and when we prepare the packaging material, they are coming along with the packaging material. And then another category is plant or plant parts or vegetative propagules are there. So these are basically the 4 major pathways, and we have to be very careful while processing the exotic material which are meant for the import purpose. So these are the steps which we follow for the processing of the germplasm or the samples which are coming inside the country. First, we go for the visual inspection. Then in visual inspection, we look for the symptomatic seeds, and then we carry those seeds for the specialized detection techniques in the laboratories. And then if we get any nematode, then we go for the identification. And here in identification, we see whether this nematode is present in our country or not. And if it is not, and if it is quarantine important nematode, then we take the decision that material will be rejected and it will not be allowed to be enter into the country. So every material we are getting from outside the country that goes through these 3 stages. So first stage is visual inspection, where we observe the seeds, open the seeds in the joint inspection lab, and we look for the shriveled, discolored, malformed seeds or the seed galls, then soil clods, plant debris, and if it is vegetatively propagated, then we look for the leaf spot lesions. And if roots are there, then root lesions, galls, and then necrotic areas on the roots are looked during the visual inspection. And these respective samples are carried for the further inspection in the laboratories. So if you see the truly seed-borne nematodes. There are some nematodes which are able to withstand the very low moisture present in the seeds, and they can survive inside the seeds and can be carried along with the seeds from one place to another place. And when we resow these seeds in the new areas, this nematode get activated and they start infecting the growing plants or the plant parts. So these kind of the nematodes are this can be spread very easily because we are not able to see the nematodes which are microscopic in size, and we cannot directly observe them under the microscope which are present inside the seed. We have to follow some steps. We have to allow them to come out in the water, and then we can confirm that this seed is infected with the nematodes. And sometimes few seeds are very hard, and we need to cut upon the seeds or crush the seeds to facilitate the nematode movement. And then suspension is observed for the presence of nematodes under the microscope. So these are the symptoms of the seeds. This is the very common nematode, which is seed gall nematode, Angina triticide. These seeds are the modified wheat grains, and you can see how the seeds are modified by the nematodes. So this can be, these are called as seed galls. By visual inspection, we can separate it out. But if anybody is not having knowledge about this nematode which is present inside this, then this can be carried to the other areas. And this is the bean seed infected with the Ditylenchus dipsaceae. The whitish part is the nematodes which are coming out when the seeds are teased. So this is how the seeds are collected. During the joint inspection, we collect some seeds, and then these seeds are soaked in the water for 24 to 48 hours. These are the wheat seeds, then rice, the chickpea, onion, hing seeds, monk fruit seeds. These are the seeds which are randomly taken. And sometimes we know the symptoms, so we look for the symptomatic seeds. But if in some cases we don't know the symptoms of this nematode, so we have to take the samples randomly for checking. So after 24 to 48 hours, we have to take this water. We have to separate water, suspension, and the seeds, and then we have to observe this suspension under the microscope. So we have to take this just a minute.
प्ले ऑप्शन वाज देयर, ये हाँ यहाँ पर है, नहीं आ रहा है.
But it is not. I have selected this laser pointer. That's why I think it is not coming. Hiding.
अभी आ गया.
So this is how we have collected the suspension. We have separated the water suspension from the seeds, and we have taken it in one beaker, and then we have to allow it to settle down for the at least 1 to 2 hours. Then nematodes will settle down, and we have to carefully remove the upper water so that the nematodes will be left in the very small quantity in the beaker. So this is how this upper water is removed in the new beaker. And when the very small quantity remains, then we have to take it into the fresh cavity block. This is called cavity block, having a small cavity. So this suspension is taken in the cavity block so that it can be easily observed under the microscope. So this is the stereosome microscope. If you find the nematode inside this water suspension, then we can go for the observation under the compound microscope, because here we can only see whether nematode is present or not. But when we need to identify the nematodes, we have to use the compound microscope. So in this suspension, if nematode is present, then okay. Then if not, then we have to follow the another procedure. That here we can see these seeds are soaked in the cavity blocks, and if it is not, we are not getting the nematode in the suspension after 24 to 48 hours of soaking, then we have to cut this seed open so that we can observe it under the compound microscope directly. So here the individual seed is cut into the two parts using sharp blade, and then the slide is observed under the microscope. Here you can see that the nematodes are here near the pointer. You can see it is small curved structure is there, and this blackish part is the seed. So under microscope we can zoom it. This is the 10x power. We can go up to the 100x, and we can identify the nematodes after doing the proper fixing and the mounting of the nematodes. So this is the process for the hard seeds, or if we are not able to get the nematodes. So this is stereo zoom microscope, then compound microscope, and under the compound microscope we can see the nematode very clearly. So this nematode is we are getting in the rice, that is called white tip nematode of rice, Aphelenchidis baeccii, and it is identified using the pointed mucro, which is present at the tail tip. And another important feature is this median bulb, which is squarish in shape. So this kind of the diagnostic keys or particular characters we look for identification of the nematodes. Then another important pathway that for the nematode introduction is sometimes we get the soil along with the seeds. So soil, as you know that the most of the nematodes are the root endoparasites or root parasites. So they live inside the soil, and along with the soil there is a risk of introduction of the nematodes. So whenever we find any soil along with the seed material, then we separate it and we process the soil by different technique for isolation of the nematode. So for processing of the soil, first we have to mix the soil in the water so that water suspension, soil suspension will be developed, and then we have to pass it through the series of the sieves, and we have to take out the content from the individual sieves, and we have to observe under the microscope, and if nematode or nematode stages are present there, then we can look for that. So these are the different sizes of the sieves: 60 mesh, 100, 325, 400, 500, and this is the micron or the pore size. So here we can see the video. So this is the soil we got from the seed samples, along with the seed samples. So we have to take it in the water so that we can mix it well. So when the soil plots are completely dissolved, then we have to, till then we have to mix it. So this is small quantity, little quantity of water. We can add it. More water to it. Then this soil suspension, we have to pass through the series of the sieves that I have told you. This is 60 mesh, 100, 325, 400. So stake of the sieve is taken, and over that we have to pour the soil suspension. This soil suspension then will pass through the different sieves, the coarse particles or the stones which that will remain on the upper sieve, that is 60 mesh sieve is there, and the small particles along with the nematode small stages and eggs will go and settle on the another sieve, that is 325 or 400 mesh. So 1 or 2 times we have to pass it. We have to avoid any escape of the nematode in the session.
क्या हुआ लीलियम वाले का राकेश?
Subhash, please mute. Then we have to use the water so that the nematode, we have to incline the sieve so that this content over the sieve will be gathered at the lower end, and then that suspension is then taken in the beaker for observing. So on the every different sieves, that is 60 mesh sieve, then 100, 325, 400, different stages of the nematodes can be collected. Like for example, very fine sieve is there, then we can collect the eggs on that. That is 400 to 500 mesh sieve. So this is the last one on which very fine soil particles come, and the nematode, small nematode juveniles and egg stages can be taken out from the sieve. So water, large quantity of water should be used to so that the soil will be washed away and nematodes will remain in the clear suspension. So only water can be taken. We have to wait for a few seconds so that the soil will remain. Then this suspension, again, we have to keep it for the 1 to 2 hours. And when the nematodes are settled down in the beaker, then we can transfer it to the cavity block and we can observe it under the microscope. In the same way, we can process the soil which is collected from the root samples. So sometimes we carry the samples from the sites during the post-entry quarantine inspections. That also can be processed using this. And sometimes we get the soil along with the bare-rooted plants, along with the roots. So that soil also processed using the same way. So these are the different stages found in the soil debris or soil clods. The juvenile stages, sometimes adult stages. These are the cysts that can be. The cyst can be collected from the 60 mesh sieve. Then these adult stages we can collect from 325 sieve, mesh sieve, and the juveniles can be collected from the 325 to 400 sieve. Then egg stages, for egg stages, we have to go beyond the 400 up to 500 mesh. So in case of the vegetative propagules, sometimes we get the vegetative propagules like bulbs, cones, rhizomes. Then we have to take few bulbs or the cones, and if it is large quantity, then we can observe a few 2 to 3 bulbs or cones, and if it is the small quantity, then we can cut the part or the piece or we can take out the soil which is attached to it, and we can process it. So this is how these are sometimes dipped as such, or sometimes we separate it out, then we cut into the pieces, and we can take out the suspension and we can observe it. Similarly, if we get the leaf or the leaf samples, then we need to look for these necrotic lesions or the spots, and this might be caused due to the nematode infection. So we take those leaves or leaf infected area, we cut them into the small pieces, and we soak it into the water. And water suspension can be observed for the presence of the nematodes. So this is for the bare rooted plants. Sometimes we receive the bare rooted plants. This is the apple plants, which are came from the European country, and you can see these roots are containing the soil adhering to it. So the soil is very important pathway for introduction of the very economically important nematode like root knot nematode, densis nematode, legend nematode, and many nematodes which are present in the soil which are harmful. So we have to be very critical while observing the bare rooted plants. So we look for the symptoms like root galls or the necrotic lesions, and we cut those roots, small roots, and we take along with the soil, and these roots are then cut into the small pieces, 2 to 3 centimeter, and this can be dipped inside the water for 24 to 48 hours, so that if migratory nematodes are there, then they will come out from the root in the water. If ectoparasites are there, then they can be easily seen in the water suspension. And if there are endoparasites, sedentary endoparasites, then we have to check them by another method, which is called root staining method. So these are the this is how these bare rooted plants come, sometimes along with the leaves, sometimes without leaves. So these are washed thoroughly, the roots are washed thoroughly so that the adhering soil can be removed, and then this soil, this suspension, this water suspension is passed through this series of the sieves, and that sieve is suspension is can be directly observed, or which can be put on the special assembly, which tissue paper mounted on the wire mesh, and that wire mesh is dipped in the water, so that it facilitates the nematode movement. So this is how the roots are washed. Then from vegetative propagules, we can extract the nematodes stages like migratory endoparasites, hemiendoparasites, then ectoparasites like nematodes can be taken out. So for migratory stages, we have to collect the infected roots, and then we have to cut it into the small pieces, and if hard root is there, then we have to macerate the root in the blender in small quantity of water, and we have to put the root on the wire mesh with mesh dipped slightly in the water, or we can directly keep into the plate which allow the migratory stages to come in the water suspension. Then if suspension is clear, then we can observe it under the microscope, or if suspension is not clear, then we can pass through the 325 or 400 mesh sieve and wash it thoroughly, and that suspension can be observed under the microscope. And in sometimes there are some endoparasitic nematodes are there, which will not come out when we actually put them in water. So we have to stain the roots. So there are basically two methods are used. this lactophenol acid fetching method, and another one is Bayer bleaching and staining method. So here the collected root plants are first made free of soil by washing them thoroughly, and then we have to cut these small pieces. And for lactophenol, 0.1% acid fetching lactophenol solution is prepared, and the roots are boiled for the 2 to 3 minutes. And it depends upon actually the root thickness. If the root is thick, then we have to give the more time for boiling, and if it is thin, then 1 to 2 minute time is sufficient. Then we have to take out the samples and wash under the running water to remove the excess stain, and then these roots are covered with the liquid phenol and allowed to differentiate. In this method, the nematode takes red stain, and this another method, sodium hypochlorite acid fetching method. Here, actually, the first bleaching, this method is also called as bleaching and staining method because sodium hypochlorite is used. Sodium hypochlorite is used for removing the clearing the root external surface. For that, 2.5% sodium hypochlorite solution is made, and the small pieces of this roots are kept for 1 minute to 4 minutes, depending upon the root thickness. And then we have to wash it thoroughly, and again we have to keep it for at least 4 hours, or preferably overnight in the fresh water, so that the traces of sodium hypochlorite will be removed. And this stock solution of acid fetching, glacial acetic acid, and distilled water is prepared, and out of that stock solution, we have to dilute that solution into 1:8 ratio with water, and we have to heat this solution for up to the boiling point, and then in boiling solution, we have to dip the roots for 30 seconds to the 1 minute. And the same way, excess stain is removed by washing, and then the roots can be directly absorbed, or this can be allowed to destain by putting them into the glycerin. So in this method, the nematode appear pink in color. So this is the Bayer bleaching and staining method which we were talking about. These are the roots are taken, and then these roots are cut into the small pieces, 2 to 3 centimeter long, and then we have to wash it thoroughly. Then we have to collect the suspension, and then we have to transfer it into the sodium hypochlorite solution for 4 minutes, and then we have to keep it for wait it for 4 hours. Then again, in the next day, these are wrapped into the muslin cloth bag, and this working solution is prepared in 1:8 ratio. This is how it is boiled over the hot plate, and then again these are taken into the washed and taken into this. This is how the nematode look inside these roots. So after that, identification of nematodes. So that after detection of the nematodes, these are need to be identified for the proper decision making, and this can be identified using morphological methods, biochemical methods, molecular methods. So for morphological methods, first step is to kill and fix the nematode. Then dehydration, mounting for the compound microscopy observation, and using the universal keys, standard keys, we have to identify the exact species of the nematode. So for killing and fixing, several fixatives are available. So for best results, we have to kill and fix the nematodes immediately. And the fixing of the nematodes allows or help to preserve the nematode in its original condition, or it avoid or arrest the post-mortem changes in the cells and tissues of the nematodes. So first we have to concentrate the extracted nematode in some small quantity of water in cavity block, and we can directly pour the hot water on this little quantity of nematode in the cavity blocks, or we can directly add the hot fixative. So these both method can be followed. This is the formalin acetic acid fixative is most commonly used. It can be prepared in 4:1 and 4:10 ratio. Then for processing, for mounting dehydration, rapid lactophenol method is also can be used, and another method, Senhouse method, is used. So for rapid lactophenol method, this is the composition of this lactophenol, and for glycerol ethanol method, these are the two solutions mostly used for the dehydration. That is 96% ethanol and glycerol, and solution to glycerol is 5 parts and ethanol is 95 parts. So for dehydration step, in this method, this is fast method where 0.5 ml of the solution 1 is taken. This solution 1, which contains the 1 part glycerol and 96% ethanol, 20 parts, and water 79 parts. So in this, the nematodes are taken in the cavity block in the small quantity, and to this we can add to this cavity block nematode from the fixative are picked and added. So this solution is taken, and then for the nematode from the fixed nematodes in the fixative, this can be picked and added to the solution 1. Then we have to keep this cavity block in the desiccator containing either ethanol, 96% ethanol, or it can be, in case of the slow glycerol method, calcium chloride can be used. So this total desiccator can be transferred at the temperature of 35 to 40 degrees Celsius, and in at least 12 hours, this ethanol will be evaporated, and the nematodes are left in the glycerol, mostly glycerol, and small quantity of the ethanol. Then to this cavity block, we have to add the solution 2, which contained only this glycerol and ethanol. So again, it is then transferred to the for 3 hours at 40 degrees Celsius, and the nematodes are left in the pure glycerol. So this is how these nematodes are, fixed nematodes are there. From here, we can pick them up, and in the solution 1, this can be kept inside the desiccator. The desiccator should be closed. Then after that, we have to take those nematode out on the slides. Then slides are prepared, and the slides are properly labeled, and after identification, the species name is written, then what kind of the nematode is there, it is mentioned, and it is kept for the further records. Then for molecular identification, this PCR method can be used using the gene specific or universal primers, and the scar markers are also available, nematode specific scar markers. Then LAMP assay can be used, real-time PCR, DNA sequencing. This area we are now strengthening, as ma'am told you. So these molecular methods are very highly sensitive, and one advantage is that this do not require adult stages for the identification. These molecular methods are highly sensitive, and this can detect the DNA extracted from the single juvenile stages or the egg stages. So for that, the nematode, we have developed our standardized protocol for the nematode DNA extraction within as nematodes are very microscopic. So we get very little quantity of the DNA, and then we go for the PCR amplification with the universal primers or the gene-specific primers. So these are the set of universal primers available for the nematodes, which are of the ITS region. Then 28S DNA fragment is there. So this primer can be used, or gene-specific primers or the nematode-specific primers are also available in the literature. We can go for it once we identify the or once we know the genera of the nematode. So after PCR amplification, we can observe using this agar gel electrophoresis whether we are getting the amplification of the desired size of amplicon or not. Then we can send it for the sequencing. If the single band is there, then we can sequence it using the outsourcing, and we can use that sequencing data to find the similar sequences in the NCBI database, so that we can have the idea, or we can supplement this data with the morphological identification of the nematodes. So this is all about the practical about the nematode, and I'm thankful to the director NBPJR and head division of plant quarantine, and all the scientists and technical staff of the quarantine division for help, and I'm very much thankful to the organizers for giving us the opportunity to share our practical experience with the trainees. Thank you.
Thank you, Dr. Gowde. Now I may request all the participants, if you have any question, you can ask from our speakers. Kavita ma'am, yes, ma'am. Dear participant, our speaker are with us. You can ask if you have any question. If you need any clarity, if you do not understand anything, please switch on your video. Any question, any clarification? If you want to shout any clarification, you can ask. Ma'am, there is one question from Dr. Devarajan.
Ma'am, please unmute.
Which species of brookid is prevalent in India?
Prevalent in India.
Yes, ma'am.
So most commonly occurring species are Calosobruchus maculatus and Calosobruchus chinensis. These are the two Calosobruchus species which are in majority of the cases very, very common, occurring all over the country and infesting lot of commodities. And the beauty of this Calosobruchus species is that it can develop generation after generation in storage conditions. So a large number of generations can per year. So 8 to 9 generations can occur in a year on the storage commodity.
Any other question?
Madam, this is Dr. Dhanavendan from Central Silk Board, madam.
Okay.
Yes. Madam, any cocoon silk, because I am working in sericulture sector. So any cocoon import or export time, any detected any insect pest, madam, or any such a thing is happened?
Unfortunately, we have not imported any insect material So
Not insect, madam,
NBPGR
the
is
cocoon
only
is
in
having some
what?
beetles infestation storage while.
Are you getting
export
some
are-
beetles infestation on the cocoon?
Yes, yes. When you are storing that, I don't know that exact which it is look like our that copra beetle like that only, madam. But if possible, I will send you
Maybe you
samples.
can send me a sample.
We are, yeah, that's why I'm asking any cocoon or importing them. Now we are reduced, but even then, any quarantine pest found in silk cocoon while transport or export.
I have no information of that, but maybe if you can send me a sample of that of that insect, you can just send me the insect either suspended in alcohol or dry mounted. I will try to identify it.
Okay, thank you, ma'am.
Yeah.
Thank you, Dr. Dhananandan. And now,
thank you.
Any other question, any participant?
Ma'am, this is Dr. D.K. Payasi from Sagar, ma'am.
जी sir, बताइए.
Actually, I am also in charge in seed production as well as certification at our stage, and in bulk quantity we are producing the seed and certifying.
Okay.
So this time we have faced some storage problem. Which fumigation method or chemical is appropriate for storage of linseed, flaxseed?
Linseed.
Yes, ma'am.
You can use phosphine, I would say.
Phosphinide.
Phosphine.
Okay, okay, ma'am.
Okay.
Right,
but you have to be very, phosphine is highly toxic. So better is you have a proper facility, facility, and then you can do the fumigation, and thereafter you can air it out so that any traces of phosphine are removed.
Right, ma'am. Right. Thank you very much, ma'am.
Any other question? Yes, madam. Malar, madam. Devarajan, ma'am.
Yes, sir. Sir, I have a question.
Yes.
Sir, so madam was telling about the brookit screening and whether which method is best. I'm asking about the screening method. Which method is best for screening the brookits?
Screening the brookits for what?
For the infestation, or whether it is to study the resistance, whether it is susceptible or resistant for that.
Depends on your crop.
So, for example, ma'am, we are studying in green gram means. So, which procedure?
Green gram means you can use both Callicebrucus maculatus as well as Callicebrucus chinensis. Better would be you use chinensis.
Chinensis, okay, okay. But
it all
here
depends,
in South India,
if you are using
Tamil Nadu, madam, in Tamil Nadu, this maculatus is prevalent. So
More
we are
prevalent.
mass culturing this maculatus only right now.
Okay. Both are quite prevalent all over the country, and they are infesting a wide range of commodities. So you can use either. If maculatus is more prevalent, you use that.
Okay, madam. Okay.
No issues.
And one more thing, madam, you are telling about that X-ray radiography, no? That that facility is whether the PG students they can avail the facility there.
We do sometimes help out students to do the X-ray, but you need to write to the director, and then once he marks it to us, then we do.
Okay, ma'am. Okay,
because this is a public organization.
Okay, madam. Okay.
He will help you. TNAU students, we are always ready to help.
Because, ma'am, why I am asking is she is undergoing one training in IR right at present. So she was asking me whether this facility, X-ray radiography, whether we can avail in PBGR.
There was one student who came to me, and she's going to come tomorrow.
I think she may be the. I think she
is it the same student?
I think she must be. Her name is Lakshmi.
Yes,
from
Lakshmi only.
entomology department.
Entomology department. She came to
She is
me
working
and
on brookits only in green gram in my crop only.
Yes.
So she
She
was
is
asking me
already
whether.
her X-ray is scheduled for tomorrow.
Okay, ma'am. Okay. Thank you. Thank you so much. Thank you, ma'am.
Thank you, Devrajani ma'am. Any other question? Any other participant you wish to ask question?
Otherwise, how are you finding the training?
Ma'am, meant to me.
I'm asking the participants.
Yeah.
Good, ma'am. Good. No problem.
Good.
Did you get some knowledge or you people? Is it find?
Yes. We are getting more about this, especially for
Will it
registration.
help to you in your
Yes,
in your work?
It is highly supportive for our research in the coming research, and now whatever we are going, that is well defined, fine tuning.
Good to hear that.
Yes.
Any other?
Daily we are updating some information, new information. It's good only.
Okay, then if there are no question, then I may propose a vote of thanks. I, on my behalf and on behalf of organizers, I thank you, Kavita ma'am and Dr. Bharat Gowde for showing practical on plant quarantine processing in of germplasm in respect of insect and nematode detection. Madam and sir, you both beautifully explained insect and nematode detection technique, different this detection techniques. And ma'am, you shown this X-ray radiography and transparency test with videography was in a very simplified manner. And sir also shown the interception of nematodes in germplasm and their identification using videography. And thank you very much, both of you, ma'am and sir. Thank you very much.
Thank you very much.
Still, any participant, if want
Thank
any
you, sir.
question, to ask any question.
Thank you, madam. Thank you, sir.
No, thank you very much. Thank you, participant.
Do you have something else now, or are you,
ma'am,
closing for the day?
Yeah, closing of this day.
Oh.
And tomorrow we'll participant. Tomorrow we will meet at 9:30, and hope you will join before 9:30, because all the time our speakers join and you people are joining late. It's not. It feels awkward. All are requested you join 10 minutes before. Thank you very much.
Thank you.
Sir, close कर दो meeting. Yes, close कर दीजिए अभी.